It's going to be on insect vectors and phytoplasmas. And we have got 7 speakers in this session, and it's my great pleasure to introduce our first speaker, Nicola Mori, and he's going to speak about the distribution and role of known potential insect vectors in flavescence dorée infected vineyards in northern Italy. So, Nicola, I would like you to come for your presentation.
Thank you.
Oh, sorry, is that Nicola? Sorry.
Okay, thank you, Christine. Good morning to everybody. It's a pleasure for me to start. I don't know how can. Okay, so this survey was done in collaboration with a group of Elisa Angelini of the National Council of Research in Vineyard. And yesterday, some of our colleagues from France and Italy showed us the importance of the flavescence dorée in Southern Europe. I confirm that. In fact, since 2019, flavescence dorée is currently the main phytosanitary issue in Veneto and other northern eastern Italy regions. In 2022, symptomatic grapevine were present in 76,000 hectares out of 106,000 cultivated. As you can see in the picture, in green, you see the grapevine growing areas, and colored with the orange and the red, the infested area with the relative incidence. Let me to show you just a couple of pictures to discuss how heavy is the problem in our region. Here we are in Verona, in a Soave grape growing area after cutting. Here close to Trento in the north part of the region. It is compulsory to cut the plants above the graph point, and after that you can remove the canopy during the season. Here in Prosecco Hills and on Cabernet in Venezia Plain. As discussed yesterday, flavescence dorée phytoplasma is a European quarantine organism, and its control relies on the mandatory eradication of infected plants and, above all, with the insecticide application against the vector. In Italy, we usually apply two insecticides, the first against the first stage, when the first stage appears, and the second one indicatively after 2 weeks, or in any case before the flying of the adults. But since 2020, epidemic outbreaks have been reported even where the correct Scaphodeus titanus control is carried out, and even where its population density was very, very low. So the aim of this present work was to investigate the spatial distribution, the FDP positivity of non-eputative vectors to understand if they have an active role in FD epidemics. We studied, first of all, Scaphodeus titanus, who's a vector of FDP from grape to grape, Orientus ishide and Dictyophora europea that are able to transmit to grape from natural reservoir, but also we investigate Aligus, Mixus and Modestus that are able to transmit the group 5 from Alnus to Alnus and to Fava, Euchelidius variegatus, that is the, as yesterday Francesca told us, the lab vector to Fava, from Fava, but also we investigate others, Ischimonus amatus, Phlogothetics cyclops, Aligidius atomarus, that are able to carry the group 5, And finally, Neolithurus fenestratus, because it's able to carry many phytoplasmas. In 2021-22, in 40 highly FD infected vineyards, the presence and the distribution of the vectors, we evaluated positioning yellow sticky traps according a line transect line sampling from the wooded part to the central part of the vineyard, and the symptomatic grapevine presence and the distribution, checking the symptoms during the August, looking around 1,000 plants along the line transect that we have considered for the traps. After that, this is the ELISA task. The total nucleic acid, both from insect and symptomatic grapevine, were extracted, analyzed, and characterized. I asked to Elisa, who is connected by web, if it is necessary to improve this part and to answer for any question about this. About the presence. Scaphodeus titanus, Orientus hichide, Phlogothetis cyclops, and Neolithurus fenestratus were the most widespread species. We found them in almost whole vineyard with a very high population density. About the distribution, according with the ecology and the biology of the vectors, we found the polyphagous species in the wooded area or in the surroundings. Instead, Neolithurus fenestratus and Euchelidius variegatus, due to the fact that above the young form lives on the herbaceous plants, such as Trifolium or Aster, we found them mainly in the central part of the vineyard. And the Scaphodeus titanus, we found it mainly in the border of the vineyards, probably due to the effect of the insecticide application and the treatments. All of the investigated cicadelles we found positive to FD phytoplasma. Mainly Scaphodeus titanus, Alligus modestus, and Alligidius hatomarius, as you can see, with a very high infectious rate. About the genotype, in the grapevine, Elisa founded M3 and M54, and we found the same map genotype only in Scaphodeus titanus, Orientus hichide, and Euchelidius variegatus. Instead, on the other vectors, no putative, we found many other genotypes. If we compare with the spatial analysis the distribution of the insects and the distribution of the symptomatic grapevines, we found a significant correlation, just to put an overlapping, and after analysis with the informatic system, with about interpolation, we found here there is the number of the positive correlation out of 40 investigated vineyards. And as you can see, we found a positive correlation with the Scaphodeus titanus, Orientus hichide, Phlogothetis cyclops, and Neolithurus fenestratus. So, in conclusion, Scaphodeus titanus confirmed that it is the main vector with the main role in the FD epidemiology, but also, if we put the results together about the distribution, the presence, and the FDP positivity, we found that Orientus ishide and Phlogothetis cyclops could have a potential role in the FD epidemiology. And about Neolithurus fenestratus, due to the high captures, the positivity to FD phytoplasma, and the not completely but partially association with the symptomatic grapevines, we have to improve to study in a deep way with a proper transmission trial to understand its role in FDP epidemiology. This work was founded by Veneto region with the project Survey on the New Epidemics of Flavichance doré in Veneto region. Many thanks for your attention.
Thank you very much, Nicola, for that presentation. We have got time for a question. The other Nicola.
Nicola, what do you think about the use of weeds inside the vineyards? They may work as a reservoir of the phytoplasmas and some insect, not Ampelophalu, strictly speaking.
Thank you, Nicola. This is a very good question. I think that in the afternoon there is a presentation about this, but yes, just to complete the epidemiology issue, we have also studied the presence and the distribution of phytoplasma, FD phytoplasma in some herbaceous plants. My opinion is that mainly the perennial one could have a role in the FD spreading. Speaking about Euschelidus variegatus or also Neolithurus fenestratus, they can acquire the phytoplasma in the grape, probably yes, Euschelidus is able to do. I don't know about Neolithurus, but in my opinion, the managing of the cover crops is very important also to reduce the spreading of phytoplasma disease.
Thank you.
Are there any further questions? Yes, up there. There's a question on the right-hand side.
Christine, I don't know if Elisa Angelini wanted to say something.
Yeah,
in the
I
web.
will ask her now. Yeah.
Okay.
No,
After that.
sorry,
okay.
there is a question. Sorry.
Okay, yeah. So my question goes in the same direction. For example, Aligos species or Orientus ishide, you also found on the borders, let's say on the bushes or even trees.
Trees, yes, mainly trees.
Did you check those borderlines for phytoplasma infection?
Yes, and honestly, I don't know exactly the results about this, but we found phytoplasma, FD phytoplasma in Quercus, Alnus, Halder, and I don't remember, Robinia, and I can provide you the complete list or this. It's quite complicated. Yeah. It's quite complicated. We thought that only for 1 year. Instead, now we see that also for Flavichance doré is not so easy in some cases to study the epidemiology of this disease.
Okay, thank you.
There is another question up there for the lady in black, and then Asunta has got a question or a comment.
I have two small questions. One of them is when you are isolating DNA from the insect, did you crush the insects or did you keep them as a whole in a buffer?
I kindly ask to Elisa to answer, but I guess we crashed.
Crashed.
Okay.
Second question is, did you do any experimental transmission trials by the putative insects that you mentioned already? Did you do any experimental transmissions?
Yes, we are going to do in this here.
Yeah.
Yesterday, Scaphodeus titanus was born in Italy. And so when I returned from Oman, we are going to do with this year with the Neolithurus fenestratus in collaboration also with Fabio Quaglino, Phlogothetis cyclops, Euchelidus variegatus, and we compare with the performance of Scaphodeus titanus.
Okay, thank you.
That was born yesterday.
So I would also like to give persons online a chance to ask the questions. Are there any questions online? Are there any questions online? If not, then there is a question, a last one, here on the left in the audience.
Thank you, Dr. Nicola. I have two questions. One of them is repeated. The first one, the rule of the epidemiological rule of Scaphodeus titanus in the Flavescens durae is confirmed. But you collected, or this vectors collected from different geographical zones, right? Do you think there is a differences in the transmission capacity between the populations or the specimen collected from different geographical zones or not? And what do you think about the South Mediterranean area?
About, sorry?
South Mediterranean region. We don't have, until now, we don't have Flavescens durae, what do you think? What is your recommendation to survey or in searching of these vectors?
Okay, in the south of Italy, Scaphodeus titanus is present. Okay. I'm sorry, I'm not able to understand, sorry.
Okay, my questions, and yeah, to present, we have no Flavescens durae.
Yes.
In Middle East or North Africa. Do you think this or such vectors are available or not, and why?
Okay. Oh, this is a good question. I have no idea about, because some are from Palearctic region, other are from Oriental region, other from Nearctic region. So I mean that they are able, those of come from Palearctic and the Oriental region. I think yes, it is possible. Quite difficult for those vectors, no, or putative that comes from Nearctic region. I don't know if I well understood the question or.
Okay, it's not only a last question, but it is a little answer. I can add something to your question. As far as I do is in Veneto, but we collaborate also in other regions, until Abruzzo, that is Middle Italy, and we have Scaphodeus in that region, but it was never detected infected by any kind of group 5. That is better say group 5 unless you have finer identification with the type described by us before and by French people after, because the system, especially the real-time used, do not discriminate among the different strains. And this is also can be a problem in the future. Claiming Flavescens durae is not, but this is a different story, it's a laboratory story. But for the southern part, I think the critical point is if the insect vector or putative vector infected incidentally arrive in this country and are adapted to the area, this can be a problem, because it is easy for them to be adapted in a little spot where the environment is okay. For example, Scaphodeus titanus is delivering the eggs under the bark. So if you buy infested cuttings, you can carry it. It happened in Italy once. Nothing happened, fortunately, about epidemic. But you know, you have to look at several points. And also the adaptability. In other countries like Chile, they verified by informatic tool that if Scaphoideus incidentally arrives, it can spread because the climatic situation are fine. They are not traveling so far, but we are traveling them. This is what I can say. I think I did everything. I have no question for you.
No, just to say, sorry, Christ.
Nicola for the presentation, and I would like to invite the next speaker. It's Agnieszka Zwalinska, and she's going to talk about potential insect vectors in a different group of crops, of wheat and maize crops, and moving from Italy to Poland.
Good morning, everyone. My name is Agnieszka Zwalinska, as I was presented, and I work at the Adam Mickiewicz University in Poznań, Poland. And today I would like to share some data of the monitoring of leafhoppers and plant hoppers from the wheat and maize fields located in Poland. So phytoplasmas infect variety of plants, including high-value crops. And they impair the plant growth development, leading often to the plant sterility, as it is observed in, for example, wheat, and also all the plants that develop a full phyllody symptoms under the phytoplasma infection. And as you probably well know, the three groups of insects are vectors of phytoplasmas: leafhoppers, plant hoppers, and psyllids. And the identification of phytoplasma vectors is a crucial step in the understanding of the disease epidemiology. So cereals are a major food source for human diet and animal nutrition, and wheat is consumed worldwide. However, in the temperate regions of world, it's a major crop. And European Union, before the Brexit, was the global leader of the in wheat grain production. Now is the second largest producer of wheat after China. Maize is also a major part of diet of many regions of the world, such as Americas, Africa, and it's important in livestock nutrition. And the European Union is a fourth global producer of maize grain. In Poland, more than 50% of cultivated area is sown with cereals. So the Poland is the third largest producer of cereals in the European Union. The wheat is dominating in a cereal structure, in a grain production, and maize, fodder maize is still, the area under fodder maize cultivation is still growing. Maize and wheat are susceptible to a certain phytoplasma strains, such as, for example, 1B phytoplasma strain, which is affecting maize and causes maize bushy stand in Central and South America and India. The typical symptoms of a disease are reddening, shortening of internodes, and lower grain formation. Vector is Adalbrus maedis. Maize can be also infected by phytoplasma from group 12A, subgroup 12A, And the typical symptoms are reddening of plants, abnormal yield development, and the rapid desiccation of plants. However, the stunting is not observed in this disease. The disease is called mate redness, and it's reported mainly in Balkan region of Europe. And the vector is Raptulus panseri. And wheat is susceptible to 1C phytoplasma strain, which causes a wheat blue dwarf, and plants show severe stunting, producing extra additional stems from the plant basal part, and sometimes they show also a bluish green leaves from that is the name of the disease. It's a problem in arid and semi-arid regions of Western China, and the disease is transmitted by Psammothetic stratius. So in Poland, we also found a few maize plants infected with 1C and 1L phytoplasma strains, which showed this bushy appearance, this stunted growth, but they were just single cases, and also symptomless infected wheat with a 1C phytoplasma we found in Poland. So there is a major concern about the future, that the phytoplasma diseases outbreaks will increase due to the global warming that helps to survive winters to the insect populations. And the aim of this study was to monitor wheat and maize plantation in selected region of Poland in order to indicate potential new phytoplasma vectors and detect early phytoplasma diseases outbreaks. Insect captures were carried out in two terms, reflecting the two developmental stages of crops. So tillering and stem elongation phase of wheat, and 7 unfolded leaves and 9 nodes phase of maize. The collections were carried out in southern Poland. Insects were identified based on their morphological features under stereomicroscope. Excuse me. And phytoplasma detections was carried out by DNA extractions using animal tissue kit, followed by nested PCR assays and PCR product sequencing. Phylogenetic trees were generated in order to identify phytoplasmas, and identification was confirmed by pair-to-pair comparison of the obtained sequences with the reference phytoplasma strains. So in total, in wheat fields, 748 leafhoppers were captured, and they belonged to 7 different Cicadelidae species. The most abundant was Zyginidia scutellaris and Psammothetic alienus, and third was Empascapteridis. However, the Zyginidia scutellaris and Empascapteridis were numerous only in an early developmental stage of wheat, which was in November, and in a late spring, in May, these insects were not found so numerous anymore in a field. From maize, in total 899 insects were captured. They belong to 9 species of Cicadelidae family and 2 species of Delphacidae family. And again, Zyginidia scutellaris and Psamothetics alienus were the dominant component of the population of insects. And the third insect species was the Leodelphax stratelius. 49.9% of insect captured in maize and 1.74% of insect captured in wheat tested positive to phytoplasma. In both crops, wheat and maize, 1C phytoplasma strain was dominating, and in total 52 phytoplasma sequences derived from insects were classified as 1C phytoplasma strain, and only 5 sequences were identical to the 1B Phylodiphytoplasma, Rhapsid Phylodiphytoplasma strain from Poland. So only 3 insect species were positive to phytoplasmas, were carriers of phytoplasmas: Psamothetics alienus, Zyginidia scutellaris, and Leodelphax stratelius. And this is the first report of phytoplasmas 1C and 1B infected Zyginidia scutellaris insects and 1C infected Leodelphax stratelius. So the results showed the presence of phytoplasma of subgroup 1C and 1B in insects feeding on maize and wheat. The insect infection rate were around 2% and 5%, which suggests the moderate risk of phytoplasma disease outbreak. Zyginidia scutellaris and Psamothetics alienus feed in large numbers on wheat and maize, suggesting that they may travel between these crops and potentially transmit the phytoplasmas. Zyginidia scutellaris is a new pest in Poland. It showed up in Poland only in 2018. However, this is the insect feeding on mesophyll, so this is the open question if this insect can actually transmit the phytoplasma. It shows some probing behavior before proper feeding, so maybe accidentally it can this way acquire a phytoplasma strains. So far this massive showed up of Zyginidia didn't correlate with bringing any new strains of phytoplasmas to Poland. This insect was only a carrier of the, let's say, native strains of phytoplasmas to Poland. So I want to thank to the National Science Center for funding my research, and also to Institute of Plant Protection for support and facilities. And thank you for your attention.
Thank you very much for the presentation. Are there questions? Yes, there's one question from Wolfgang, and we will have time for a second question.
Thank you for the presentation. I have just a question for understanding. So these insects overwinter as egg state or as adult state?
This insect, excuse me?
How do they overwinter, the insects, with the potential vectors?
Psamothetics alienus overwinters are as eggs, in eggs stage, and about 2 others, I'm not sure.
But the background is, what is the source of the phytoplasma infection? Perennial weeds or?
I think so, yes. The insects have to feed on some weeds in between the crops are available in fields. So they usually they prefer these young stages of wheat and other grasses, but this is not always available, so they travel into the margins to feed it, to get additional
there you
feed.
find infections, because you have to have infected plants first.
Excuse me?
that the new adults acquire the phytoplasma, you need to have infected plants first.
Yeah, sure. They acquired from somewhere, yes.
Is one very short question and short answer because we need to move on.
Have you tested maize and weeds at the same time, while you are testing the insects? I mean, did you find group 1, B, and C phytoplasmas in maize or wheat?
As I told at the beginning, yes, in maize there were a few cases of positive maize plants, but in different regions and different periods. That's why I thought to first screen the insect and the level of the infections of insects in fields. And the wheat is sometimes also infected, but we don't observe the symptoms.
But you detected the phytoplasma in wheats?
In wheat, yes.
Okay.
Yeah, thank you very much for this interesting presentation. Thank you. And now we have got to change. The next presentation is going to be online, and it's going to go by Wayne Murray, and he is in Jamaica and talking about phytoplasmas in palm trees. Can we move to the online presentation, please?
Thank you very much. Good morning, good evening, good afternoon, good day to everyone. I'd like to thank the organizing committee and the people and government of Oman. Also want to thank the IPWG coordinator, Professor Sultan Qaboos, for providing the opportunity to make this presentation on the assessment of proportion of populations of Plexus, Scudus, and Equus macrospringi carrying the lethal phytoplasma in Jamaica. Also want to thank my colleagues, Erica Helmick and Professor Brian Bader from the University of Florida. Pests and diseases are known in the coconut growing producing areas in Jamaica. The lethal yellowing disease is one such. It's caused by phytoplasma, bud rot caused by Phytoptera catsuri and Phytoptera pomibora. You have dieback. You also have anthracnose caused by Coleototricum glorosporidase. Various leaf spot diseases such as the Drusularia and Pistolotia leaf spot diseases. Of course, in terms of pests, you have the ambrosia beetle, red palm mites, scales, and gall mites, all affecting coconut production in Jamaica. The lethal yellowing disease is the most important disease for us in Jamaica. The lethal yellowing disease devastated in the 1970s 8 million Jamaica tolls, and in the 1990s up to 2000 have destroyed many coconut trees. We have conducted various research on the pathogen itself and also on the epidemiological spread of the disease, and have found ways to grow coconuts here in Jamaica. This is how the disease spread in the fields, destroying every coconut tree in its wake and causing a severe reduction in the coconut production in the country. The disease generally exhibits yellowing or drying of the leaves as the third stage of the symptoms, The first symptom you would see is premature nut fall, necrosis of the inflorescences, yellowing of the leaves, and the last stage, a kind of a fall off the tree, and that is called the telephone pole. The management strategies, which are important for the decrease of spread of the disease in the coconut growing areas, are what is exhibited on your screen. And the package itself is important, and it's important in order to be able to deal with disease. 1, 2, 3, 4, 5 in that order should be carried out when managing the disease. The disease is caused by, as I said before, phytoplasma, which is found in the phloem of the tree. This is what caused the death of the tree, and of course it is important to be able to control the spread of this phytoplasma, which is done by a vector, and this vector, as it is known, is Haplexus crudus, found first in Florida in 1980-84, and it have not been proven elsewhere in the world, but certainly based on what we have found phytoplasma in the body or saliva gland of this insect, and therefore it is fair to say that it has the capacity to transmit the disease. Our method of collection of the insect is insect collection by aspiration, insect collection by sweeping nets, and of course, whenever we sweep, we make sure that what the insect we really want, that's what we collect. Those that we don't want, we actually release them back into the ecosystem. So we're very, very careful where that is concerned. And of course, the last picture on your right of the screen with the net is a transmission trials that we have been conducting in the fields to prove which of the insect or insect vector, potential insect vectors that we have found, which one is actually transmitting the disease. Potential vector that we have found is Eccleosmaca springi. This insect is located in several areas and has the potential to transmit disease. We have certainly found the phytoplasma in the body of the insect, especially in the salivary gland. More work is needed, and we are doing the work to prove that this insect is actually a true vector of disease. The method we use to identify this insect as a new species would be cytochrome oxidase subunit 1 and also the 18S rRNA gene, along with external features, morphological features that distinguishes, looking at the genitalia, etc., head, thorax, to distinguish the insect and also the wings. For identification of the insect as it relates to phytoplasma, we also do this work as well after DNA extraction. So it works both ways, DNA extraction to tell if this is the right Haplexus crudus or the Eccleosmaca springi, and also meaning there is no variation or there is not a new species, and also variation or determination, I should say, as it relates to the phytoplasma detection. So we screen a number of insects. This is just one set of data. We have a large set of data because we have collected many insects. And if you go on Zootaxa, you will see the many publications that we have released and have been published in Zootaxa from our insect collection. So Eccleosmaca springi and so forth, Sedusa, Algubini is one such new species that we have discovered. Haplexus crudus we have found. We also found Haplexus crudus, which is different from the Haplexus crudus. I found an Haplexus which is different from Haplexus crudus. It's called Haplexus phonicus. And so we have published this information as well. So for up to May, and we started in January, up to May of this year, we have gone 3 publications so far, and these are all new species that we have discovered here in Jamaica. Only here in Jamaica you can find so far, based on all the data that we have looked at, surveys that we have looked at and have conducted, even here in the region. These are insects that we find here in Jamaica. The last one is an insect that we found in Trinidad and Tobago. Thank you very much for your attention.
Yeah, thank you. I believe Wayne is online. Wayne, can you hear us? No, I just get a sign that he is not online. So I presume there's also won't be any opportunity to questions. I suggest to email him the questions and contact him that way. So I think we can move on to the next.
Or you can ask Assunta, she's here. Yeah, you know about this stuff, eh? I know. This is your project. Okay, so we'll move to another date palm with Maria, and also she is online.
In the name of
Mariam
God.
is Gaib Zamhariri.
Good
Okay.
morning, everybody. It's my pleasure to talk about my research in this meeting. Today, I want to talk about my study on epidemiology of Escherichia yellows disease of date palm in Iran. At the first, I would like to explain the importance of date production for Iran. Iran is one of the leading date producing and exporting countries in the world. Iran ranked first in the world in terms of the area on date cultivation, but after Egypt, ranking second in production and in the export of this economic product is in the 7th place. The origins of date production and use in Iran go back to about 4000 BC. Today, more than 400 types of date and various kinds of date-based food are consumed by the Iranian people. Date palm plants, like other plants, affected by different pathogens, but a new disease with symptoms similar to palm lethal yellowing was noticed in early 2030 in Khuzestan province in several date plants in Iran. Infected trees displaying symptoms of Escherichia yellows and varied in the incidence and severity of yellowing. Other symptoms include tiny narrow leaves and leaf drying. Similar symptoms could be observed in the ornamental palms. The in silico analysis of 6 DNA RFLP pattern from Phytoplasma positive date palm indicate that 2 different Phytoplasma most closely related to clover proliferation Phytoplasma, group 6, and ash yellows Phytoplasma, group 7, associate with Escherichia date palm symptoms in Iran. Before the disease observation in Iran, we have found a report from the Phytoplasma disease in Egypt that the symptoms of this disease was very similar to those we observed in Escherichia yellows Phytoplasma disease in Iran. In this country, the agent was detected, the Phytoplasma, but unfortunately, the group of the Phytoplasma was not detected. In 2080, similar disease to to Escherichia loss of date palm in Iran observed in Masqat. Disease shows dryness of young and mature leaflets and leaves, but despite of the phytoplasma agent in Iran, in Oman, a phytoplasma related to subgroup D of group 2 was reported as an agent of this disease. In the same year, this disease has been reported from Saudi Arabia, but despite of the Oman and Iran, the phytoplasma agent related to group 1. Due to the detection of some phytoplasma infected samples from Iran and difference of phytoplasma reported from date palm in Iran with other countries, we decided to study possible insect vectors in my country. In this order, insect samples were collected from symptomatic and asymptomatic date palm trees in infected orchards located in 3 provinces, including Khuzestan, Bushehr, and Kerman in my country. Potential insect vectors collected by different methods, including sticky yellow scar traps and butter traps. Among the different methods of sampling, the most successful method were shaking insects of plants and using a sweeping net. The collected insects, which comprised Heteroptera and Ochenorincha, were sorted and identified. The identification of Heteroptera species was performed by Professor Attilio Carapesa from University of Palermo in Italy. For molecular analysis of infected phytoplasma, total nucleic acid was extracted from collected insects, and the detection of phytoplasma was done by nested PCR method based on 6DS rRNA gene using the universal phytoplasma primer set P1 cysteine followed by primer set R6CF2N R2 in the collective putative insect vectors from date palm groves. At first, we tried to identify the insect samples through the collected insect samples, the specimen of different species of leafhoppers and hemipters identified. They included Ravona iranica from family Dictyophoridae, Tropidocephala, Prasina, Somatitix alianus, and Impossible decipiens that related to Cicadelidae. Since the identification of leafhoppers to the species level require the examination of the genitalia of the male samples, the identification of the collected female sample will down in the family level. He recognized some of them and identified them as Bagrada sp. related to Pentatomidae family, Istalcoriza ventarilizi related to Pentatomidae family. Also, he identified some of bugs as Nicius graminicola related to Ligidae family. Some of samples identified as Omanocoris versicolor and others recognized as Remadorierina anolipis. The collected samples, the samples related to the species Erizocoris ventrialis, Omanocoris versicolor, and Tropidocephala parasina was phytoplasma positive. The sequencing and molecular analysis of Tropidocephala prassina species resulted infected with phytoplasma related to Candidatus phytoplasma tripoli, that is a species related to group 6 subgroup A, while the phytoplasma positive of other species is not related to this group. Phylogenetic analysis confirmed that the infected insects are related to group 6 and are close to phytoplasmas infected palm in the Iran. Tropidocephala prassina, known as an endemic species in Iran which feeds on herbaceous plants, can be considered as a probable vector for palm leaf streak disease, since it was found to harbor one of the 2 phytoplasma associated with the disease. Other species that was collected from the palm streak yellows disease garden and was positive to phytoplasma infection was the species Staricoris ventrialis that related to Pentatomidae family. Also, this species was not infected with phytoplasmas that cause streak yellows disease in palm, but infected of this insect to phytoplasma was important for us because newly we have some reported that Pentatomidae family could be vector of phytoplasma disease. As reported before, 2 families of hemipters, including Tingidae and Pentatomidae, could transmit phytoplasma. Haliomorpha is one of the members of the family Pentatomidae, could transmit prewinkle phytoplasma in Japan, Korea, and China. And Stephanotis typical is the family member of the Tingidae that could transmit the coconut decline phytoplasma in some. In newly study in Iran, it has demonstrated that Ceranotide pallidus could be transmitted a phytoplasma related to seed pod abortion disease of soybean in Iran. These results and results from other studies could say that the hemipter family should be considering as new vectors in phytoplasma disease, especially in Iran, because the diversity as hemipter in Iran is very high. At the end, I thank you for all of them for attention to my presentation. Especially, I thank you from the meeting organizer, Professor Bertaccini and Dr. Asubhi, to let me present my results online. Thank you of all you.
She's in line? Okay. Yeah, she's in line, eh? Okay, so let's go to the second one with Mariam Ghaib Zamhariri about studying in soybean and insect vector. So let's make that question together at the end of this speak.
Everybody, it's my delight to present my work today. There is result of my study on soybean bud proliferation phytoplasma insect vector. Soybean has been cultivated in Iran since 1939 and at present is critically important oilseed plant in my country. Soybean bud proliferation and seed pod abortion disease was firstly observed in soybean in Iran in 1985. The symptomatic plant produced non-ovary mature brown pods and reduced yield. After 35 years research, it was verified the consistent presence of Candidatus phytoplasma trufoli in the symptomatic soybean samples. This phytoplasma could be transmitted by grafting. Interestingly, before the detection of the agent of disease, it was observed that the presence of caton shatter bugs, that's scientifically named Chironotide pallidus, was in the infected fields, and control of it could prevent the disease spreading in the infected fields. In order to study the vectors of this disease, possible vectors collected from symptomatic fields including aphids, leafhoppers, thrips, and bugs. And just the Chironotide pallidus related to Hemiptera myride specimen was infected to phytoplasma and showed 100% significantly to the phytoplasma of seed pod abortion in soybean. The collected insects fed on healthy 3 months old seed-grown soybean plants. The myriads were split in 2 groups. Group A was used for nucleic acid extraction and PCR, and Group B was used for transmission experiment. Myriads boys without phytoplasma infection were obtained from hatched eggs produced by insects raised on uninfected soybean in greenhouse conditions. Uninfected myriad nymphs were inserted in homemade cages with 2 mates on 2 mates on 1 mate on average, containing 40 symptomatic soybean plants in the field for 20 days. A question: the development of symptoms was then monitored. Soybean plants that were experimentally infested with myriads that had fed upon symptomatic plants confined in field cages showed disease symptoms 45 to 60 days after inoculation with myriad bugs. Previously has been reported 2 families of Hemiptera, including Tingide and Pentatomide, were phytoplasma vectors. For example, adults and nymphs of the brown marmorated stink bugs could transmit the Palvonia phytoplasma, or Staphylinitis typical related to family Tingide could transmit another phytoplasma in coconut palms. In conclusion, we could tell that seed pod proliferation and seed pod abortion disease in Iran consistently associate with Candidatus phytoplasma trufoli and could transmit it by Chironotide pallidus in Iran. These findings are helpful for development strategies for management of this disease in my country. Thank you once again for the meeting organizer, especially Professor Bertaccini, Dr. Assohi, to let me present my research results online. Thank you very much.
Okay, any questions? She is online. Yeah, here.
Okay. So I hope she can hear me. I don't know. Thank you for the presentation. In one of your slides, you showed the acquisition feeding. what was the final transmission rate?
Hello, Mariam.
Hello, everybody.
Yes, did you hear
Would
the
you
question?
please say, yes, I'm ready. I'm ready. I'm ready.
Shall I repeat the question?
Thank you. Yes, please.
Okay, so in one of your slides, you showed the cage with the acquisition feeding. And
Yes.
I guess the next step was to transfer the infected insects to healthy plants. What was the
Yes,
transmission rate?
At the first we have collected samples from the insect samples from the healthy fields, and then in the greenhouse I'm feeding them on the infected ones. After demonstrated the insect acquisition, the phytoplasma, we transmitted it in healthy phytoplasma for detect the ability of transmission of this insect.
Yeah, and what was the incubation time and what was the final transmission rate you found?
The acquisition was around 50 to 30 days on the greenhouse condition, and the rate of the transmission was very high. I could told that more than 90% of the plant was infected.
Okay, thank you.
Thank you. Okay, I think we start with Mahdi. And Mahdi gonna talk about the simsim in Arabic and phytoplasma, the insect vector. Yes, Mahdi. Mahdi also is from Iran.
Okay, very good morning, everyone. Actually, this is the first time that I'm attending the IPWG conference, and I would like to thank the conference organizer, Sultan Qaboos University, and the IPWG for giving me the opportunity to attend the conference and also for this presentation. Now I start my presentation that is entitled Molecular Characterization of the Phytoplasmas Associated with Sesame Phyllodes Disease and Identification of its insect vector in Kerman province of Iran. Yes. As you know, sesame is one of the most ancient oilseed crops, and we say that is the chief oilseed crop in the world because of its extraction process, good stability, high oil and antioxidants, and also for the drought resistance. And as you know, sesame is cultivated in more than 80 countries, mostly in the tropical and the subtropical area of the Asia, Africa, and America. Can you see? You can see in the map also. Presently, sesame production is more than 6 million tons per year by the more than 1 million hectares, and the most top countries for sesame production, as you see, are Myanmar, India, China, Sudan, Tanzania, Ethiopia, Uganda, and Nigeria. But in case of Iran, sesame is cultivated in Iran for more than 42,000 hectares with the 43,000 ton production per year. And in case of my province, Kerman province, is the third rank for sesame production in Iran by the more than 8,500 cultivated area. As you know, among the different biotic and abiotic stresses on sesame, phyllodes is one of the most important diseases in tropical and subtropical area. And when we reviewed the literature, we found that Cerculifer hematoceps, Orosius albicinctus, and Hishimonos fissitus have been reported as insect vectors for sesame phyllodes associated phytoplasma in the world in different countries. And in my state, in my province, Kerman province, phyllodes is the most important and most destructive disease of sesame. And there is no solution, there is no management program for this disease. And there was a gap for this research. So we started to define a project for biological and molecular characterization of the phytoplasma associated with sesame phyllodes, I mean for etiology and also for the insect vector identification. Here you can see the map of the Kerman province that we collected the samples from different 9 cities in Kerman province during the flowering season. And we did the standard works, DNA extraction and the PCR assays by using the 16S ribosomal RNA gene and some other genes like SEC-A, SEC-Y, and RP genes for multi-locus sequence analysis. And also we analyzed the sequences by the specific and the common softwares and programs by these primers that you can see. And so also we collected the leaf hoppers from the sesame fields. That was it was included the Orosius albicinctus A, Cerculifer hematoceps, and M. pasca decipiens. This was the most popular leaf hoppers that we collected from the infected fields in my province. And then we tested the leaf hoppers for phytoplasma association, phytoplasma infection, and also we tried to use this insects for the transmission trial in the grasshopper. The inoculated seedlings, we used the seedlings for the inoculation, and the inoculated seedlings were kept in the greenhouse, and we searched for the symptom expression, and also we did the PCR and sequencing for the sequential identification of the associated phytoplasma. So here you can see the symptoms and the severity and the incidence of the disease in my state, that the disease incidence was varied from 5 to maximum 100%, and you can see some fields that totally are infected by this disease, and the highest incidence was observed in one of the area, we called it Rudbar Junub, that is in south of the province. Here you can see the symptoms. There was mostly witch's broom. Here you can see witch's broom and proliferation and seed pod malformation. Here you can see the healthy branches in the flowering stage, in the pod seed stage, and this one is the infected branch. Also here, we can see the healthy branch in the seed pod stage and the infected plant with the different samples, with the different symptoms, virescence, phyllodes, and pod malformation. And also one of the symptoms was the seed pod cracking. Here you can see the result of PCR detection. We did the PCR amplification for the symptomatic and asymptomatic samples for the leaf hoppers, collected leaf hoppers. But we found the phytoplasma, we got the specific phytoplasma band in the symptomatic plants and also in Orosius albicinctus and the Circulifer tenellus, but not in the healthy plants or asymptomatic plants, and also we couldn't get the amplification in Emposca leaf hoppers, as you can see here. When we analyzed the sequencing data, we found that the phytoplasma in these symptomatic plants and also in the Orosius albicinctus and the Circulifer tenellus was identical, and all of them were belong to the group 2, subgroup D. Then we use the multi-locus sequence analyzing. We use the SEC-A, SEC-Y, and RP genes for the further identification and confirmation of the results. Then we found that the results also was similar, and the associated phytoplasma in the plants and also in the leaf hoppers was belong to the same group. So when we did the transmission, we found that the Orosius albicinctus and the Circulifer tenellus could transmit the phytoplasma, and we found the little leaf and the stunting symptoms, not like very similar to the field symptoms. Here was the stunting and the little leaf. But when we did the PCR, we could detect the phytoplasma. We did the sequencing, and it was confirmed that the same phytoplasma was transmitted by these leaf hoppers. As you know, these leaf hoppers has been introduced as the vector in some of the other countries, including Turkey, India, Thailand, and also in central provinces of Iran. These insect vectors has been introduced as the sesame phyllodes phytoplasma, but the phytoplasma was not exactly similar. It was different, that I will show you in the next slides. Now I'm going to conclude that the phytoplasma associated with sesame phyllodes in Kerman province of Iran is belong to 16S ribosomal 2 subgroup D, and the leaf hoppers Orosius albicinctus and the Circulifer hematoceps could transmit the associated phytoplasma to the seedlings. So we are introducing these two leaf hoppers as the insect vectors for this phytoplasma. And also with, in the other research work, we found that many of the vegetables and the oilseeds and the ornamentals and alfalfa were the infected and host of this phytoplasma. So it is very important, and many of these plants have the overlap cultivation time. So it will be a great threat for horticulture and also for the crop plants in the country. Here I collected the data and reviewed the literature that you can see. We have 4 distinct ribosomal groups of the phytoplasma: group 1, group 2, group 6, and group 9, that till now have been reported in association with the sesame in 9 different countries that you can see in the map from different countries, and which phytoplasma groups has been reported. As you can see, in Iran, we have group 2 and group 6 that were associated with the sesame phyllodes, but in my state, in the Kerman province, we found only group 2. So the last sentence I would like to propose that I think it is very important. We have many different phytoplasma associated with sesame, and so it is very good that if we could define or design a mega project, worldwide international mega project for study the etiology and transmission and epidemiology of the sesame phyllodes disease. Thank you so much.
Okay, Nico.
Yes, thank you. Thank you for your presentation. How many days you leave the insect feeding on the plants in the trials, transmission trials?
How many leafhovers?
Yeah. No, how many? How many days?
How many days?
Days. How many days you leave the insect feeding in the plants?
On the healthy seedlings, yes?
Yeah.
Yeah. Actually, we release the leafhovers on the seedlings for 15 till 1 month. 15 days for 1 month. And we searched the symptom expression.
The insect didn't die?
We didn't kill the insects.
Okay.
We didn't remove the cap.
You corroborate the presence of the phytoplasmas in the insect put in transmission trials?
Sorry?
You corroborate the presence of the phytoplasmas in the insect used in transmission trials?
No, we didn't do it.
Okay.
No, we didn't do it.
Thank you.
Thank you. Okay, last question here.
Just a consideration, we, I think, publish or at least say already that also in sesame there is a seed transmission of phytoplasmas. Do you consider this possibility or not?
We didn't try this.
Okay, thank you.
Okay, thank you so much.
Now we move to insect vector of phytoplasma and soybean with Karian. She's from India and she'll start now.
Good morning all. My name is Kiran. I'm from India. I have been allotted 5 minutes. I won't take more than 5 minutes. The coffee is waiting. I'll be talking about insect vectors of phytoplasma in soybean fields and discovery of Mystes dorsalis and Exitianus indicus through feeding medium assays. So in India, 2 species of phytoplasma, specifically Candidatus aurantifolia and Candidatus australicea, have been predominantly associated with phyllodia and witch's broom diseases of legume plants. Now we have identified the similar species are infecting the weeds in the surrounding fields. Now the rich fauna associated with these weeds is responsible for further spread of the disease. So we tried to understand the diversity of cicalidae insect vectors in soybean field. We collected around 500 insects over the span of 2 years, and we identified them based on morphological characters and COI gene barcoding. COI gene barcoding is particularly important to enrich the databases so that the non-entomologist people can also identify insect accurately with sample PCR assays. So, these identities were confirmed with phylogenetic analysis, and we observed that Exitianus indicis and Mycetes dorsalis were abundant in all seasons in our collection. So we wanted to check if these insects are able to transmit the phytoplasma, and the first choice was a biological transmission assays. We have seen presentations discussing about biological transmission assays. I'll quickly run through what is a biological transmission assays, where you rear the insect family, phytoplasma-free insect family, and then you allow those insects to feed on the infected plants, allow them to acquire the phytoplasma, transfer them to the healthy plants, and allow them to feed over there, and check these healthy plants for symptoms. Now this is very lengthy and tedious process, and it can be very challenging for a person, non-entomologist like me. So we decided to go for an alternative assay, which is a feeding medium assay. So in this assay, we took 1.5 ml Eppendorf tubes. We cut the base of this tube with, and the base was sealed with cotton plug to allow the air passage. Now the tip of these tubes were filled with 200 microliter TE sucrose, and we sealed with the sterile parafilms. The live insects were put inside these tubes, and they were allowed to feed this artificial diet for 48 hours. Then after 48 hours, we sacrificed these tubes, and we checked the presence of phytoplasma in insect body as well as in feeding medium. Through nested qPCR assays. Now the number for these assays runs in 600s, including Exitianus and for Exitianus and Mycetes dorsalis. So they were checked spread throughout the 20 qPCR plates, and this graph shows the PCR efficiencies of those qPCR assays. So we confirmed the presence of phytoplasma in the feeding medium, and we hereby confirm that the Exitianus indicus and Mycetes dorsalis were able to inoculate the phytoplasma in the artificial feeding medium. When we check the presence of phytoplasma in insect bodies, we see a constant present throughout the year in insect bodies, and similarly in the next season in 2021. But we saw a seasonal pattern in feeding, inoculative ability of these insects in both the seasons. So here we propose that the PCR assays of artificial feeding media are more efficient for high number of insect samples and vast diversity studies in epidemiology. Thank you. Yeah, I'm sorry. I'll thank IPWG. Yeah, just a minute. I'll thank IPWG for allowing me to present my research over here. I'm thankful to DBT, UGC, NCRB, and NMPB for project fundings for this project, as well as the other research going on in my lab. And a special thanks to TSBAS for funding this trip. I would also like to thank my colleagues Bhavesh Tivrekar, Vipul Thorat, and of course my guide Dr. Amit Yadav. Thank you.
Thank you.
Yeah.
Thank you. We have one question because we have a coffee break and people here, they give me eyes, so I have to go. Okay, one fast question. No? Internet, they have anybody online? Want the questions? Okay, nobody. Thank you. And I would like to thank everybody for the speakers. And please, you have a coffee now, and you have to back in 11 o'clock. Thank you.
Thank you, Prof. The coffee break is ready, please.
Not coughing. We have to consider we are late. Okay, we definitely start. We hope the rest of the people will enter soon. The first speaker of this session, the session is Epidemiology of Phytoplasma-Associated Disease. The first presentation, titled Jujube Witch's Broom Disease, Bacteria that Drive the Plants Crazy, is given by Jidong Lee. Jidong, please.
Thank you, Professor Batasini. Good morning, everyone. I'm Jidong from Henan Agricultural University, China. My topic here is about Jujube Witch's Broom Disease, also called Zaofeng disease in China. Actually, it's a review about the Jujube Witch's Broom Disease research. First, Jujube, in Chinese we call it Zao. It's a traditional fruit in China. It could be eaten fresh or dried. According to historic research, it has more than 7,000 years cultivation history. So it's a very traditional fruit and have a very big industry in China. Jujube Witch's Broom disease caused by phytoplasma, also in Chinese called Zaofeng disease. Zao means Jujube, and Feng illustrated means crazy, mad, or uncontrolled. One thing I need to clarify here is that in Chinese, different characters may have the same pronunciation. Feng means uncontrolled, crazy, or mad here for Zaofeng disease. And you may notice that in our article, our group leader, Professor Feng, he is a very disciplined, motivated, excellent researcher, scientist. He is not crazy. The Zaofeng disease, or Jujube Witch's Broom, was first reported in the 1950s, according to literatures. And nowadays, it's widely distributed in China. Actually, in 1951, Professor Ji, he first reported it as Zaofeng disease. According to recent research, every cultivation area for Jujube has witch's broom diseases, so it's getting more and more serious. And according to literatures, it's also reported in India, Japan, and Korea. The Jujube has nowadays cultivated in more than 40 countries all around the world, but Jujube witch's broom only reported in India, Japan, and Korea. The sample terms of Jujube witch's brooms, including witch's broom, are also shoot proliferation, and phyllodes, also leafy flower, the yellowing, growing, staining, and yield loss, and the infected tree will die within a few years. Same sample terms will appear regardless of the cultivar or geological areas. At first, the Jujube witch's brooms were believed to caused by virus, and in the 1970s, it was named as MLOs. In 2003, it was named as Candidatus Phytoplasma zizifi. According to the rDNA, it's the 16R5 group, subgroup B. The Jujube witch's broom phytoplasma are transmitted by phloem-feeding insect vectors, such as mainly leaf hoppers. It also can be transferred by graft and not by pollen, seeds, or root contact or soil. Despite for jujube, many other plants are reported as plant host of candidate to Phytoplasma ziziphus, the jujube which bloom Phytoplasma. The Chinese jujube and the world jujube, also called soil jujube, and the Indian jujube, these are genus ziziphus, and other fruit trees such as apple, peach, sweet cherry, plums, date palm, and some garden trees and some other crops. In fact, with Phytoplasma, the jujube trees may be the biochemical and physiological effects, such as photosynthesis, the phytochemical parameters and related genes were affected. And the mineral elements, such as K, Mg, Fe, will be in fact affected by the Phytoplasma infection. The diseased trees also show the variation in anatomic structures, such as branchwood vessels, lumen diameters, and the phytohormones, such as GA, jasmine acid, and auxin, and salicylic acid, were also affected by the Phytoplasma infection. All the literatures marked with green are reported by our group. As for in recent years, more molecular methods are applied in the jujube bloom research, such as these two experiments are pursued by our group. The first is we infect the health trees with graft and sampled in 6 stages. The other is treat the in vitro diseased seedling by tetracycline, so that eliminated, to turn it healthy, and sampled in 3 stages. And then we do the RNA-seq and the eye-track proteomics on these samples. Some genes and pathways are revealed as various, different expressed, especially those about jasmine acid synthesis. Actually, there are a lot of works on like this. These are maybe all the reports, papers on the genes and pathways involved in jujube Phytoplasma interaction. They have SSH, qPCR, RNA-seq, and proteomic to study the corresponding pathways or corresponding genes. Many have focused on the logs, the TCPs, and the matchbox genes, which were proved have responded to the Phytoplasma effectors. And the genome of the jujube Phytoplasma, there are 2 jujube Phytoplasma genomes reported now. I have just showed one for here. It's reported by Professor Wang in Shandong Pomology Institute in 2018. Our group has also tried to get the genome of jujube Phytoplasma. by disintegration, centrifugation, and pulse-field gel electrolysis. The genome of jujube phytoplasma is 750K BP. It's a circular chromosome. If we have the genome information, make it possible to further study on virulence effectors. In our group, we have reported the one is 早风 6. 早风 6, it could induce dwarf and small leaves, and we prove it interacts with TGP7. Actually, it's a SAP11 homolog. And we made a TGP7 mutant by CRISPR. The mutant have a similar phenotype with the 早风 6 transmitted seedling. The other one is 早风 3. This has been accepted by phytopathology recently, will be published. It induces leafy flower and also shoot proliferation. It's a very interesting thing, because we have confirmed that it interacts with matchbox TFs and TGP7, also with TGP7. It's a very interesting effector. Another group in China, Professor Sun Jun from Anhui Agricultural University, have also reported similar effectors. She named them as SGP12, interact with TGPs, and the other is SGP3. It interact with MAS. Actually, SGP12 are homolog of 早风 6, and SGP3 is a homolog of 早风 3. For diagnosis, actually in the field you can observe by sample-term observation, you can distinguish the disease trace with the health trace. The phytoplasma also can be detected by DAPI stain or by PCR. Recent years, more efficient and methods have been developed. We have all-in-one dual CRISPR assay by CRISPR 12 methods for the phytoplasma. It has been pre-printed on the Authora. You may read it on Authora. This method can be used in the field directly, not by the PCR machine. Do not have many complex utensils, so that it's maybe very quick and easy to perform. For the disease control, the first we believe is the resistance breeding. Because jujube has many cultivars in China in different areas. According to the Chinese fruit tree book, there is more than 700 cultivars. So different cultivars may have different susceptible or different resistance. And even in the same cultivar, there will be difference for the resistance in different trees. So the screen of the resistant jujube cultivar or different or super trees are very important method for the resistance breeding. The other method is for the application of antibiotics, such as tetracycline. But in field application, it's still not so efficient. Comprehensive control methods are also be put forward for the jujube witch-broom disease control. They have 4 parts of work: diagnosis and quarantine, phytoplasma-free seedling cultivation, and the orchard management. Then, if there were disease trees, how to treat them. For that part, you may cut off the diseased branches like a surgery for the tree. Or if the tree is too heavy, infected, just cut it off. That's a brief review about the jujube witch-broom phytoplasma research. For the future prospects, we think that the one thing is that we may maybe get more genome resources to study the properties of the phytoplasma. Actually, our group have also have a brief phytoplasma genome assembly. We are still mining the data in it, so it's not published. And we have assembly for the 3 jujube witch, assembly them with the other reported genomes. So it's very interesting that they have a little difference. So we think that maybe we could get more genomes resources to start the involvement of the different strains, between the different strains. Maybe it will get more information about how to. And the other thing is the effector protein structure, to why they have the function to cause the disease symptom. Maybe the structure is very important. The other is the interaction of phytoplasma with the plant and their insect vectors. There are 2 kind of, the first is the effectors and the membrane proteins. It is reported on wheat blue dwarf phytoplasma disease. There is a iron silencing suppressor, and we have screened all the candidate jujube witch-broom phytoplasma effectors. Maybe there is some iron silencing suppressor in the GWB phytoplasma genome. That's all I want to say about jujube witch-broom phytoplasma. Thank you for your attention. Thanks a lot for all the comments.
Thanks, Zhidong, for your presentation showing the situation of this disease in China. I had the opportunity to see it, and it is very serious. We are at time for maybe one question. You have a question from the audience? Mm-hmm.
So thank you very much for your presentation. I have a question because you talk about the Chaofeng protein is interact with TCP7. Did you have any idea about its sequence? Did you make alignment to SAP11 because the SAP11 is prototype for TCP binding and interaction?
Pardon?
Do you have any? Do you know the Chaofeng by TCP7? Did you compare the sequence Chaofeng to SAP11?
Chaofeng?
Chaofeng, I don't know, your protein, Chaofeng. Number 6, yeah?
Oh.
Maybe I speak, don't speak Chinese, but you Southing.
6 or 3?
Yes, because they interact to TCP 6.
Yes.
6, no? And did you know that the sequence of southing is compared to SAP 11?
Compared to what?
SAP 11.
SAP 11?
Yes.
Yeah, they are homologs.
There's a homolog, that's okay. But you don't know the why they degrade TCP or they just buy the mechanism of this.
TCP 7. It actually is TCP 7.
Yes. They degrade.
We screened it with all the TCPs in Jujube.
You know only it's interact, but don't know the mechanism of this yet.
Okay, I think you can further discuss during lunch or during break. Even if there are questions from the online, I hope not, because we have no time. Okay, a question? Very quick, please.
Thank you. It's not question, but rather maybe comment. Like, I have one question, but you said the jujube witch broom, it's from the resistant type phytoplasma.
What type?
This is from the wild type or from the cultivar type?
Wild type.
From the wild type. Because in Oman also we have jujube plants. So as per my knowledge, the highly resistant comes from the wild type. So
where it comes from, we still don't know. But wild type jujube and cultivated jujube, they also have the same disease.
So for the management control, I mean, it will be very good to collaborate with other countries and collect some jujube accessions. What do you think from Oman
All
also?
right.
Yeah, thank you so much.
Okay. No time for any more questions. Thank you very much for your presentation. The next presentation is origin of isolated cases of flavescens dorée in northeast of France, search for reservoir plants and insect vectors in semi-natural habitats near vineyards. And the presentation will be given by Arthur Oriol.
Hello, do you hear me? Maybe it's better like this, I know.
A bit more. More microphone, thanks.
Here? Yes. Right. I have to put it manually? I have to put it? Okay. Okay. So good morning, everyone. I am Arthur Oriol from Bordeaux, and as my gentle colleagues already talked to you about flavescens dorée, it will be brief for me to repeat some things about this disease. So it's an epidemic quarantine grapevine disease caused by phytoplasmas from group 5 and subgroup C and D, and it's infected most of southern Europe viticultural areas, but unfortunately there is no curative treatments, but only preventive means by prospection of grapevine stocks in the vineyards, and when a symptomatic grapevine is infected by flavescens dorée phytoplasma genotypes, it has to be removed, and then the wine growers may have to apply some insecticide treatments in order to counter and eradicate its vector, which is Scaphoideus titanus. It comes from North America, and it's a grapevine specialist, and it's considered as the main vector of flavescens dorée phytoplasma genotypes. So for the flavescens dorée epidemiological cycle, when Scaphoideus titanus is present in the vineyards and when Do you see? Yeah, the point? Yeah, okay, great. So some epidemic flavescence doré genotypes are compatible with Scaphoideus titanus, and they can lead to flavescence doré outbreaks. And then in an horizontally way, Scaphoideus titanus can transmit these genotypes from grapevine to grapevines. However, even if Scaphoideus titanus comes from North America, the origin of flavescence doré phytoplasmas are European, and they are naturally harbored by European alder, Alnus glutinosa, which is considered as the main natural flavescence doré phytoplasmas plant reservoir. On these trees, some of other leafhoppers, such as the genus Aligus and Orientus ischidae, which are oligophagous insects, they can acquire on alders some of the flavescence doré genotypes and then transmit them from alders to alders. And then if these trees, alders, are located near vineyards, it can sometimes happen with a low frequency transfer, a transfer from these genotypes from the alders to the grapevine. So these insects are considered as alternative flavescence doré vectors. And some other plants in some other European countries, are known to be infected by other flavescence doré genotypes, and as the same way, for alders, this tree hopper, Dictyophara europea, can acquire on clematis some genotypes and transmit it with a low frequency transfer to grapevine. So this is an overview of the FD epidemiological cycle, and that can lead to outbreaks if Scaphoideus titanus is present in the vineyards. However, we also can find some isolated flavescence doré cases that don't lead to outbreaks. And our study, the question of our study was, is the origin of these isolated cases of flavescence doré. The main hypothesis is that from the wild environment to the grapevine, there are probable transfer of these genotypes by alternative vectors and from the known reservoir plants. So we wanted to look for the origin of these isolated cases, first by looking for some other and potential reservoir plants, and then alternative insect vectors. The experimentations took place in northeast of France in two viticulture regions originally free from flavescence doré, and some isolated flavescence doré cases have been found since 2029 up to now. On each of these sites, we surveyed and sampled some woody plants and vines located around the vineyards, or in the vineyards sometimes, and to look for some reservoir plants. We also captured Ochenorican insects either by installing some yellow sticky traps in the vineyards or in the vicinity of the vineyards, or with a classic net beating technique to capture alive insects to perform transmission assays. And then I identified every leaf hoppers. So on the samples of plants and insects, we perform a classic DNA extraction and then sequencing and of the map gene to find the phytoplasmas from group 5, C, and D, and other phytoplasmas. But I'm more an insect guy. I'm not good in molecular biology, so sorry in advance. And we discovered, like on the amount of a plant we inventoried and we sampled, we only found the alders to be infected by flavescence doré-related phytoplasmas with a mixed infection of group 5C. No other plants have been found to be infected, neither the clematis, hazelnuts or willows, or other plants that are known to be infected by these genotypes. So it is our only potential reservoir that we detected. On the insect side, I would not have the time to go in details, but we found a lot of leafhopper species and infected by some different groups of phytoplasmas. And then now I'm going to show you some sites and a hypothesis for each site. Firstly, in Champagne, we have these vineyards here located near some rivers with European alders, and there is a lot of Scaphoideus present in a really high densities population. And so the main hypothesis say the same. Is there a transfer from the European alders to the grapevine that may have caused, for example, this isolated FD case? I don't know if, yeah, okay. It's a M50 genotype, so an epidemic one. So the alders were found to be infected, and we didn't find any insect positive to flavescence dorée phytoplasma. The aligus, which are known to be alternative vectors, weren't infected too. So for this origin of this case, we have no clue, and this case remains unknown, so we need further surveys. Now we're going to the Alsace region. Here there is no Scaphoideus titanus in this vineyard. However, a M38 genotype have been discovered in 2019. So the hypothesis stays the same because the European alders are really near the vineyard. The alders were found affected, and we found a lot of species infected by M38 genotype, such as known alternative vectors that were infected in a high infection rate, Orientus aligus and Lampotetrix nitidulus. And we also found other species infected, but these species are not known as alternative vectors. So this case is due to a probable transfer from alders by alternative vectors. However, we have to perform some transmission assays on these other species. And then the last site I will show you, if we come back in Champagne, where there is a lot of Scaphoideus titanus present in the vineyards, located near the Marne River, with a lot of infected alders. It's still the same, a transfer from the alders to the grapevine. The alders were infected, and we only found one alternative vector infected by M38 captured on alders. So maybe we miss some of other vectors, and it's a probable transfer from alders by alternative vectors. However, we also found some Scaphoideus titanus infected by M38 in the vineyard and in the European alders, since they can't eat well on alders. So we may draw more hypotheses. Maybe it's a propagation from grapevine to grapevine by Scaphoideus titanus with a low frequency transfer, or is Scaphoideus titanus able to transfer, to acquire phytoplasmas on the alders and transmit it to the grapevine. For answering this question, we performed some acquisition assays. So is Scaphoideus titanus is able to feed on infected alders, acquire flavescence dorée phytoplasmas, and transmit it to grapevine? First, we needed some FD infected alders. For that, we capture Orientus ischidae on the infected alders. We put them on healthy alders, and then we waited the phytoplasma to incubate in the alders to get some infected ones. Then we put some larvae, sorry, it's an adult photo, but we put larvae of Scaphoideus titanus on these alders, and then for 3 or 4 days, we let them acquire phytoplasma. And then we put them on latency on Vitis vinifera, and then in transmission to broad beans. We get, and then we were looking at the FD symptoms, and we performed molecular diagnostics. We found infected broad beans, so we can conclude that Scaphoideus titanus is experimentally able to acquire on alders and to transmit flavescence dorée phytoplasma, but we need to repeat this experiment. So to conclude, if I come back to the cycle that I presented you before, here we can maybe draw a new cycle of isolated flavescence dorée cases in northeast of France for the genotypes M38 and M50. So we still have Scaphoideus janus on the vineyards, and maybe it can acquire, it can be the source of the M38, for example, cases that we found in the vineyards. And then, we also have probability of these vectors to be the source of the isolated cases. And then since we found a lot of new vectors to be infected by M38, maybe they can be considered as additional alternative vectors. Unfortunately, we didn't find some other reservoir plants, so we need to go further. And maybe Scaphoideus janus is able to acquire on the alders and transmit to grapevine. So we still don't really know, but we may find some new ways to, new plants to prospect and new insect to test to complete the cycle, or try to. Thanks for all of my colleagues and all of the local partners in Champagne and Alsace, and thanks for your attention.
Thank you. We have time for some questions. Yeah.
Thank you, Arthur, for the nice talk. You showed that Scaphoideus is able to acquire the phytoplasma from alders experimentally, but did you look for Scaphoideus on alder or other plants on the border of the vineyards? Did you find some species or adults of Scapho?
We focused on the infected on alders. So we found them on alders, but they are present in the wild compartment in other plants. Mm-hmm. But the density population of Scaphoideus janus is huge, so maybe they just arrived there by accident.
Mm-hmm. But on alders they were present.
Yeah.
So, yeah. Okay. In all these regions you investigated?
We have some sites I didn't present where there were no alders in the vicinity of the vineyards, like they were maybe 1 or 2 kilometers away, and we never found other infected plants. So what was your question? Sorry. That was your question?
No, it was like if you found Scaphoideus titanus on alders in the adjacent to the vineyards in the different regions you investigated.
Oh, we only found in Champagne, in the different sites of Champagne. But we only found when the vineyard was present, well located near.
Mm-hmm. Okay, thank you.
We have another question on the back.
Yeah.
They cannot hear you from home.
Nicola,
okay.
We need a microphone.
Sorry.
Thank you, Ali. It's a very interesting point. But which stage did you find in alder about Scaphoideus titanus? Adults or youngs?
No, only adults.
Only adults.
Yeah, and because we captured them like in late July, and it was only adults. But maybe there are some abandoned grapevine located near alders, and they may, but no, adults.
Okay. Are there any more questions?
I would have a question from the online. I'm online from Italy, Elisa Angelini.
Welcome,
Hello to
Italy.
everybody. And my question is not really a question, it's a comment, because actually I would like to say that in our region, Veneto, we have a lot of Scaphoideus everywhere in vineyards and a lot of alders which are infected, but in all these 20 years, we never found the same genotype that are present in grapevine in alder. So in our cases, it means that we don't have a real transfer from the wood, from alders in this case, to the vineyard by means of Scaphoideus. Just for giving some hope to people, you know. Thank you very much.
Thank you.
Do you have an answer or no?
Okay, that was a question, but I just I heard like badly, so maybe if you.
Elisa, can you repeat, please?
Yeah.
Yes.
Please.
It was not really a question. It was just a comment to say, can in our region, in Veneto region, in Italy, hopefully we don't have the same situation as all the strains of Phytoplasmas flaviceus that are in alder are not present in grapevine. So it does mean that we don't have an active transfer by Scaphoideus titanus in the vineyard, even if we have a lot of Scaphoideus titanus here.
Yeah.
Thank you very much.
Thank you. Yeah, but maybe an accident is possible. Yeah.
Okay, thank you. Some other question? Yeah. Wait, just behind you.
Yes, I have a question. Have you tested your insect DNA sample by using like a plant barcoding marker genes?
Sorry, can you repeat, please?
So have you tested your insect DNAs using a plant barcode marker genes so you can see your insects field on what kind of plant food source?
No.
No, okay. I think it's a good idea to do it because you can find the plant reservoir. Yeah, it's easier because otherwise it's you randomly to check a plant which one is infected. But if you test insect DNAs and using the plant barcode marker genes, it will be easier.
Yeah, but I only. I was not like I have not the time to perform some molecular biology, so it's my colleague that I think a lot, and they are way more better than me on this field. So I couldn't really answer you.
Okay, thank you. It is time to move.
The next presentation is epidemiology of pear decline in orchard without vector control in southwest Germany, and it is given by Wolfgang Jarausch. Thank you, Wolfgang.
So, good morning. We have to wait until the presentation appears, hopefully. Okay, the title has been introduced. Thank you, Asunta. So I can go up, maybe I save time. Background is that pear decline is a devastating phytoplasma disease in pear in Europe, but also in North America, but I will talk about our European situation. It is caused by Candida phytoplasma peri and transmitted by different pear psyllids. It has been a quarantine disease in Europe until recently. Now it's downgraded to a non-regulated quarantine disease, but it's still important economically. And it's in the past decade or a little bit longer, it's spreading dramatically in our region in southwest Germany, where we're talking about, but also in adjacent countries like in Austria and the Middle European part in general. And we assume that it's due to the reduction of insecticides in the past years. And this is in view of the new policy of the European Union. They, with this Green Deal that they want to take in place, we will miss any synthetic insecticide on PER to control the psyllids as psyllid pest, and we have to develop new control strategies, and these have to be in accordance with this new policy. And in Germany, we lived for a long time with the disease, but nobody really cared about the vector situation. We have no good data for that. And in respect to these changement in policies and treatments, so we had the chance by different projects which we have at the moment to analyze the situation and the actual situation in our small region. So we have 3 different psyllid species which are recognized vectors of the disease. 2 of them, Capsula puri and Capsula pericula, are polyvoltine species. They can have 3 to 5 generations per year, and they entirely live on PER. And they have one winter form which is developing in late autumn and overwinters on the PER tree. And then in this period, the phytoplasma can multiply in the insect, and they are the generation which is of highest risk for the transmission of the phytoplasma in early spring, late winter, early spring. That I will focus on. We have a 3rd species, it's Capsula perisuga. It's an univoltine species which recently has been described by Monika Riedle-Bauer in Austria, that it's a vector, and that she considers it as an important vector in her region. And this makes a host plant change. It's from summer to winter. This species remains on conifers, and in the same period as the other 2 will transmit these species as well. In early spring transmits the phytoplasma. And the picture below, I should have placed it on the first slide, gives you an idea about the importance of the disease. All the red trees are infected, and you maybe have seen the poster I have also on this topic. And we tested all trees, that's almost 100% infection. And with climate change, now with the drought in summer, we have also a lot of losses of trees. So it's really becoming dramatically. And in these kind of orchards, 9 of them, we captured the psyllids. And this was done by the beating tray method, and as I said, focused on this overwintering generation in 2 periods, the periods of risk, I would say, in late winter, early spring, for 3 years, that is February, March in our region, and when the new overwintering generation is developing in late autumn or early winter, November, December, we tested as well. We also checked the population densities by estimations of psyllids per branch. We tested all psyllids which we captured individually via protocol with a 69 well plate, so we could have a huge number of individuals tested. And then finally we applied quantitative real-time PCR to estimate the concentration in the individuals and normalize the data with an insect gene. So coming to the results, I hope you can see it. Then on the left-hand side, you have the data from the late winter, springtime for every year, and on the right-hand side, you have the data from the new generation of these overwintering forms from late autumn, early winter. And if you just follow the arrows, I don't want to go into much details. Capsula puri always has lower infection rates than Capsula pericula, and we tested more than 1,000 individuals, around 1,000 per season, so we can conclude something out of it. And we have a constant increase. So the next generation formed in autumn has a higher infection rate, about 1 to 5 for Capsula pericula, and these overwintered, and in the next spring we tested other individuals, but they have 2.6 and more than 5% for Capsula pericula, and it still increased. And in the next step, when the new overwintering forms are built, established, we have 4.4 with Capsula puri, which is the highest value we found, and 8.6 or 7 for Capsula pericula. But then in this spring season, I've also added new data from March, which is not in the abstract. Capsula puri is dropped down to 0.6 or below 1%, but Capsular pericula, on the other hand, is still increasing above 9%. And I also have to mention this data, Capsular perisuga, we found everywhere, but we found only one single individual. And just to show you the message in a trend, the infection rate of Capsular puri, we have some old data, is around 1 to 2%. It's a little bit, looks like an increase, but I consider it's more or less stable. We have see the variation we have. But the increase in Capsular pericula is important, in my opinion, because we did not have reports that Capsular pericula was present at all in the region. So this we reach now about 10%. And this is dramatically because we have high population densities from these. And if you just look on the blue and the red bars, Capsular puri is the population and in blue, and red is Capsular pericula. The scale is not always the same for the 3 different years in springtime, but you should have a take-home message. You have either a big blue bar or either a big red bar. So you have a sort of separation of these two species, and you can have an important population of Capsular puri and almost non-pericula, and vice versa. There are only a few orchards where you have a substantial amount of both species in the same orchard. But if you look at the infection rate of these, in these typical orchards where we find both, and we looked for these 3 years always in the same and check the infection, you still have more infection in Capsular pericula, even if it's in a lower presence compared to puri, like in this orchard from Neustadt, and vice versa. On the other hand, on the right-hand side, this orchard Ilvesheim, you have normally high populations of Capsular pericula, but the few Capsular puri also are infected. But there's always 2 to 3 times more in Capsular pericula. That are the PCR test data, but that doesn't signify that these individuals are infectious. For this, we applied quantitative PCR to check the titer in the individuals, which we found positive. So it's 14 for Capsular puri and 45 for pericula, which we could analyze. And the data are expressed as phytoplasma per insect cell. And there's a huge range of data you get. I grouped them into 4 classes. Class 1 is less than 1 phytoplasma per insect cell, and then you have 1 to 10, 10 to 100, and a few individuals had more than 100. If you give you a comparison to other published data, these 100 phytoplasmas per insect cells are in the range of other published data. So finally, from experience with other systems, I conclude that in the class 1, below 1 phytoplasma per insect cells, we don't have infectious individuals. They just acquired the phytoplasma, or they did not reach the salivary glands. So this accounts for 60% of, in both species, of the individuals we tested. And only 40% can be considered infectious. But in absolute numbers, it's just 6 in Capsular puri and 17 in pericula. So it's even always more. And though these are the conclusions, so we find all 3 psyllid vectors in our regions. And we found high populations of both polyvoltine species, and from this high population of Capsular pericula are new for our region. It's a species which is normally found in northern parts, in northern Germany and the UK, and not so far south. And this is, as I said, a new aspect. And as I repeat, that it has always 2 to 3 times higher infection rates than Capsular puri, which has been considered as the main vector so far. And this seems even to increase from year to year. And only 40% of these positive individuals are considered to be infectious, but absolute numbers, it's even always 3 times more for Capsula puricula. And what I didn't mention, we had also transmission trials with these species, and they both transmitted to test plants. So they are vectoring the phytoplasma really in our region. And the main conclusion out of our work is that Capsula puricula seems to be the factor which is responsible for the increased spread of per decline which we observe. On the contrary, and also on the contrary to the situation which is reported for Austria, Capsula purisuga seems not to be a risk for the spread in our region. And this is the end. Thank you for your attention.
Thank you, Wolfgang. We have questions from the audience for Wolfgang. Okay.
Thank you very much for your interesting talk, Wolfgang. If I remember right, I mean the previous knowledge was Cacophylla piri was the most common insect and also most effective vectors in Europe, whereas piricola was more abundant in United States. So how the situation has changed from Cacophylla piri to piricola? What is your opinion?
Yeah, I must add that there were also old reports from UK for Cacophylla puricula from David Davis in the 90s, early 90s. Now, what I didn't mention is that the hypothesis is that Cacophylla puricula is more sensitive to insecticide treatments than Cacophylla puri, and that if you apply in commercial orchards regularly these insecticides, because these both species are pests themselves without being vectors of the phytoplasma, so we had always a low amount of Cacophylla puricula which survived. But as I said, this was the objective of the presentation, that if you reduce this vector control, and we are forced to do this in future, we will have maybe an outbreak of per decline also in the commercial orchards which had so far these insecticide treatments.
Another question is very short. For the experimental transmission trials, what was the effectiveness if you compare two species, Cacophylla piri and piricola, for the experimental transmission?
For the transmission trials?
Yes.
Yeah. They were not done in this strict order, so for this I didn't present the data. They were similar.
Okay.
As it reflects a little bit the quantification. So if you had a look, the high titer individuals were in both species.
Okay, thanks.
More questions from the audience or from online? Okay, I don't see. Yeah, there is one question here.
Thank you. Very good information. Just I would like to know what are the other natural hosts for these Psilid species other than peach orchard? Is only they reproduce in peach orchards, or there's some other weed host reported in that particular area, or the other cultivated species?
It's only Pyrus species, so we have only these cultured Pyrus in our region, so there is no wild reservoir of the disease.
Yeah, I'm asking, other than peach orchard, has these Psilid species has been recorded on other, neighboring areas, or weed species?
So I didn't maybe get it,
Okay.
but if you consider the origin of the source of infection, then it's always an orchard. If this was the
Okay,
question.
so it used to reproduce in peach orchards only?
Yeah, it
Okay.
reproduce. As I mentioned briefly, it can all be, we have a lot of scattered orchards, or if you like, wild cultivated pear trees, and these are not treated for sure, and so they may be also a source of inoculum of the insects.
Thank you.
Phytoplasma.
If I can add a word, it was reported that the overwintering insects are carrying the phytoplasma to the new season. They don't need another host, at least in Italy. they can carry the phytoplasma from one year to the other, the adult, during the winter. So without alternative host. Thank you, Wolfgang, for your presentation. I think we can, there is one more question, just a moment. It's okay? No? Okay, thank you.
The next presentation is Candidatus Phytoplasma trifoli associated with Faba bin Phyllodes in Jordan, and it will be presented by Nida Salem.
Thank you. Just open. Okay, Assalamu alaikum wa rahmatullahi wa barakatuh. Peace upon you. It's my pleasure to be here in Oman in this meeting to give my presentation, which is entitled Candidatus Phytoplasma trifoli associated with Faba bin Phyllodes in Jordan. Okay, which is a collaborative work between the University of Jordan and the University of Milan. First of all, before start talking about Faba bin Phyllodes, I collected all of the data related to phytoplasma in Jordan to see how the situation in Jordan. So working on phytoplasma in Jordan started in 2002 when tomato big bud symptoms were observed on tomato, and at this time Dr. Ghandi and Fuqa, he identified or detected phytoplasma associated with this type of symptoms. So it was in 22, and in the same year also he did identification for phytoplasma associated with witch's broom on beaches. And for 9 years there was nothing about phytoplasma in Jordan until 2011 when I got contacted by Dr. Qadriye and she asked me if I can give a presentation about virus and virus-like diseases on stony fruit under the frame of the coast action. At that time I told her I am plant virologist mainly and I don't work on phytoplasma, but I'm so thankful for that event because I got the chance to meet great scientists like Dr. Sultan and Qadriya and Piero Bianco, and from there I started to work on phytoplasma. So eventually I worked on phytoplasma on grapes, and then on potato, and then on plum. And another group of scientists from a research institute in Jordan with collaboration with Dr. Khalid Lehdeb, they worked on phytoplasma on date palm in 2017-2018. And recently, Asim Abu Alloush, he joined the Piero Bianco lab in University of Milan and worked on almond and pomegranate. So the overall view of this table tell us that phytoplasma diseases are distributed in Jordan, and they can infect different hosts, including vegetables as well as fruity trees, and also they can be on some alternative hosts like Convolvulus arvensis. And in some studies, they showed the vector for these phytoplasma. However, we come now to Faba bin, because my mainly concern Faba bin. Faba bin is considered as one of the most globally important legume crops because it's very rich in protein. It has 20 to 25% protein. Also, it has carbohydrate, fibers, minerals, and secondary metabolites. It can be used for human food as well as for animal feed, and also it's very important in nitrogen fixation process. However, Faba bin is subjected to attack by different pests and diseases, including insect and fungal diseases, bacteria, nematode, and parasitic plants. Faba bin in Jordan, I was in charge to write a book chapter about Faba bin diseases in Jordan. So in 2021, in February, I went to collect some nice samples to be included in the book. And it was easy to see the fungal diseases like, for example, Alternaria spot, ascochyta spot, botrytis spot, and for example, rust, wilt, and also we saw some symptoms for nematode and some viral diseases. And also viral diseases, we could see two types of symptoms, the mosaic one and the yellows one. Mosaic symptoms are mainly caused by bean yellow mosaic virus in Jordan and broad bean stain virus, while the yellow symptoms are caused mainly by faba bean necrotic yellows virus and bean leaf roll virus. But interestingly, we saw something is different from what we saw before, and it was the phyllodes symptoms that you can see here closer, and then on this one and this one. The phyllodes symptoms mainly that we saw included dwarfing of the plant and also stunting, yellowing, and failure of the plant to give fruits. So that drove our attention to the fact that there are could be some phytoplasma associated with faba bean phyllodes in the field. In order to further go and identify or detect the causal agent associated with faba bean phyllodes, we did field surveys in Jordan Valley. And why Jordan Valley? Because Jordan Valley, it's the main growing area for vegetables in Jordan. So it's mainly for like vegetables. Because of that, we did survey and collected samples from symptomatic as well as asymptomatic faba beans. And also we collected some wheats grown in the same fields. And so you can see here, these are the symptomatic faba bean, where you can see the phyllodes is clearly, you can see it clearly here and also here. And also we collect some samples from wheats like Conobodium mural and Malva parviflora. And what we noticed in these fields, the plants were intensively invaded by leafhopper populations. So we collected leafhoppers and we divided them into 3 portions. One portion for bioassay or for transmission, and another one for DNA extraction and detection of the phytoplasma, and the third portion for taxonomic characterization. And also we collected it from asymptomatic plants. Then we proceed with the identification, doing DNA extraction using different techniques, whether from faba bean plants, wheats, and leafhoppers, and then running BCR using B1, B7, followed by F2 and R2. The expected product were observed in faba bean asymptomatic plants, as well as in Conobodium murali, in Malva parviflora, in Convolvulus arvensis, and on leafhoppers, and also on symptomatic faba bean, but it was not detected in asymptomatic faba bean. Expected bands were taken and cloned in BGMT-EZ and then sent for sequencing. For doing phytoplasma transmission, we use 2 approaches, the graft transmission and the vector transmission. In the grafted transmission, we use 2-week-old healthy faba bean plants, and we grafted them from a source which is infected with faba bean phyllodes. And after 4 weeks, you can see the symptoms of the phyllodes on the graft inoculated plant. And the phytoplasma was confirmed in all of the grafted plants in order to be sure, yes, it is the causal agent that transmitted from phytoplasma infected plant to the inoculated plants. As well as we did the vector transmission, where we kept the leafhoppers that we collected on healthy faba bean plants, and after 4 weeks we tested them by using PCR and confirmed the presence of the virus, of the phytoplasma, and then we did the cloning followed by sequencing. For the insect identification, the accurate species identification was based mainly on microscopic examination of male genitalia, and this was done by my colleague, who's the insect taxonomist, and he did the identification, as you see here, like the difference between the male and the female, and some features for the male genitalia structure. And based on this microscopic examination, the vector was identified as in Boasca decumbens. However, to do further molecular characterization for the vector, the DNA extracts that used for identification of phytoplasma also was used for detection of mitochondrial cytochrome oxidase, subunit 1 gene, and the 28S ribosomal DNA. We got the expected product by using both set of primers, and then bands or BCR products were cloned in BGMT-easy and sequenced later. Now, for sequencing and sequence analysis, we sequenced all of the products that resulted from F2N and R2 that we got, whether from faba bean or from wheats and from leaf harbors, and also from graft inoculated plants and the tested plant that used for vector transmission. And the presence of the phytoplasma was confirmed, and based on the sequence identity or blast analysis with the gene bank, it is shown that the sequence identity was more than 99.6% between the 16S ribosomal DNA and nucleotide sequence of phytoplasma that we obtained in this study compared with Candidatus phytoplasma trifoliae. That means it is Candidatus phytoplasma trifoliae. And sequences were deposited in the gene bank. For the phylogenetic analysis, the phylogenetic tree was constructed using the sequences obtained from phytoplasma in faba bean, on leaf harbors, on wheats. And as you see here, and compared with other Candidatus phytoplasma species, the 49 species, and you can see that our phytoplasma, or faba bean phytoplasma, clustered with the phytoplasma trifoliae group 6 and subgroup A. For Candidatus, for attribution to the subspecies or subgroup, restriction fragment length polymorphism was done or generated by IFI classifier, and you can see 100% identity between the pattern obtained from the Candidatus in this study and the reference strain 16SR6 group A. So, in our conclusion, our results supported that the faba bean phyllodes is associated with Candidatus phytoplasma trifoliae 16 SR6A in Jordan, and the phyllodes agent was successfully transmitted by grafting as well as by vector. In both Boasca decumbens was identified, as I mentioned, based on the microscopic examination and also by molecular characterization, and also was identified as a potential vector of faba bean phyllodes because we were able to transmit the phytoplasma from infected faba bean to healthy one. It's worth it to mention that in Jordan, there is a big survey carried out between 2018 and 2019, where a large number of specimens of leaf harbors were collected in Jordan. And it was found that this vector is widely distributed in different regions in Jordan and in a wide host range. Like 28 hosts were found to have this vector, and it's also found all over the year. That means it's considered as its role in transmission of plant diseases, and in this case, particularly phytoplasma. The presence of the phytoplasma in wheats, including Conopodium mural, Convolvulus arvensis, and Malva parviflora was really important because these host plants, they could act as alternative or reservoir for the phytoplasma. And although phytoplasma disease on faba bean has been reported in some literature, like it's found in Cuba, Egypt, India, Saudi Arabia, Spain, Sudan, Iran, and Peru. And the phytoplasma were classified into 3 groups. 16S are group number 1, 2, and 3. However, in our case, we found the phytoplasma associated with faba bean phyllodes is in a group 16S 6 group. So it is somehow different. So the story is not done yet, and we have so many things still to do. First of all, monitor the actual diffusion and impact of faba bean phyllodes in Jordan, because our study was somehow, it's like a small survey, but we have to do comprehensive survey and cover more areas. And also in-depth characterize the candidatus phytoplasma trifoliate strain associated with faba bean phyllodes, and to determine the role of plant reservoirs like Conobodium murali, Convolvulus arvensis, Malva, Barbiflora in the faba bean epidemiological pathways. And further on, as plant virologist, I'm always interested about plant viruses, because I could see some of the infected plants, they do have viruses. So I got the inspire after the yesterday presentation about studying the incidence or the presence of bacteria and phytoplasma. So I would like to do like studying further the mixed infection when the virus is with the phytoplasma. Is there any synergistic or antagonistic effect between them or interaction? And with that, I am done. Thank you. Shukran jazilan. And thank you.
Thank you. We have time maybe for one short question, if there is any. There is one.
Thank you very much for your clear presentation, Nida. Actually, it is a comment rather than question. While you are going to search mixed infection by virus and phytoplasma in the future, I would like to suggest you to test also Spiroplasma citri, which is which causes also witch's broom in the broad bean, very similar to the phytoplasmas. So especially if the broad bean fields are close to the citrus area. Yeah. Just remind that Spiroplasma citri could cause very similar symptoms to phytoplasmas on the broad beans.
Okay, thank you for the suggestion. Thank you, Doctor.
Thank you.
Okay, thank you.
The next presentation is again online because it's our colleague from Iran. So the next is soybean bud proliferation phytoplasma transmission by seed, and it is presented by Marian Zamrir.
In the name of God. Good morning, everybody. It's my delight to attend to this valuable conference, and I hope my talk would be useful for you. I want to make you a short presentation about soybean bud proliferation phytoplasma transmission by seed. At present, soybean is a critically important oilseed plant, especially in 2 provinces of Iran, including Mazandaran and Golestan. Soybean bud proliferation and seed pod abortion disease was firstly observed in soybean in Iran in 1985. The symptomatic plants produce none or few mature brown pods, and a reduced yield and significant economic losses are reported. After 35 years research, it was verified the consistent presence of Candidatus phytoplasma tripoli related to subgroup A of group 6 in the symptomatic soybean samples. This phytoplasma was transmitted by grafting. In infected fields, cotton shredder bugs that scientifically named Chronotida pallidus could transmit the phytoplasma agent. In order to study the possibility of transmitting this phytoplasma by seeds, we have collected soybean seeds from Golestan province. Altogether, 100 seeds were analyzed, 500 from symptomatic plants and 500 from asymptomatic plants. Infection was detected using nested PCR analysis, and in all samples, no virus detected after serological testing. 40 days after germination, nested PCR analyses were used for detection of phytoplasma, and phylogenetic analyses were performed with maximum parsimony analysis algorithm. Our results showed that germination percentage for soybean seeds collected from infected plants were around 76%, which was significantly different from the seeds collected from symptomatic ones, that was 98%. Only 1.8% of seeds collected from infected plants were positive for phytoplasma, and this phytoplasma is enclosed to subgroup A of group 6. Recent studies in some herbaceous crop have shown that the phytoplasmas could be transmitted through the seeds. This data suggests that phytoplasma is the agent of the seed proliferation in soybean could transmit it by seed in Iran too. At the end, I have many thanks from this meeting organization, especially Professor Bertaccini and Professor Sabuhi, for letting me to present my work online. Thank you.
I think if there are questions, there is no more time. We are a little bit behind time, so I think Mariam will understand, even if she's online, that we don't have time for this discussion.
Okay, we are moving to the next presentation, and it's phytoplasma presence and inoculum sources in carrot fields in Turkey. And the presentation will be given by Field Centrum.
Thank you. My presentation is about phytoplasma presence and inoculum sources in carrot fields in Turkey. It was our recent research already concluded. This is all kind of carrots. Especially we have natural colored and also red colored cultivars usually produced. Carrot, Dacus caruta, is a member of family of Apiaceae, is grown and consumed in almost every geographical region in the world. Turkey produces an average of 588,000 tons of carrot from more than 10,000 hectares annually, which is about 84 of production is located in Ankara and Konya provinces, located in the central Anatolia. So this is my region, and also our surveys also conducted in both regions. Phytoplasmas are prokaryotic organisms without cell walls and limited to phloem tissues, and also phylogenetically relevant to gram-positive bacteria in general. Phytoplasma infections in carrot plants are associated with symptoms such as reddening, yellowing of leaves, shoot proliferation, and decrease in the root quality. Phytoplasmas belonging to 16SR1, 16SR12 Stolberg groups in Europe, 16SR5 Almiolus group in Israel, and 16SR2 peanut witchsbrum group in Saudi Arabia, and 16SR6 clover proliferation group in USA has been reported to infect the carrots in fields. In addition to Convolvulus arvensis and Dacus caruta wild were found to be infected with 16SR12 group in Hatay provinces located in the eastern Mediterranean region of Turkey. Several studies showed that Phytoplasma groups can also be transmitted via seeds in various plant species, including carrots. Moreover, the weeds and wild plants are of high importance to understand the epidemiology of phytoplasma-associated diseases. In this study, the potential of carrot seeds and reservoir wheat species as inoculum sources of phytoplasmas that occurred in carrot fields in Ankara and Konya provinces was investigated. Symptomatic and asymptomatic carrot plants and 7 weed species in 5 botanical families were sampled from the phytoplasma-infected carrot fields and their surrounding areas in Beypazarı and Nalhan districts of Ankara and Meram districts of Konya, Turkey, between April 2018 and September 2020. 19 wheat samples were collected, including 5 Amaranthus retroflexus, 7 Scinapis arvensis, 3 Conium maculatum, 1 Biphoraridians, 1 Dacus caruta wild, and 1 Medicagus sativa, and 1 Fumaria officinalis. Symptoms such as reddening of lip tips, mosaic, and vein clearing were noted, and a few of them, while the most were symptomless. Seeds of 8, 1 locally red, and 7 commercial seeds were widely grown carrot cultivars were also obtained from the farmers during our surveys. Since the number of carrot seeds was so limited, only approximately 200 or 300 seeds per cultivar were sown on pots and containing sterilized soil and germinated. Those are the areas where we did our surveys. This is the capital, and also my university is here. This is Beypazarı and Nalhan area, and this is the Konya area. In our city, mostly the normal carrots. In the southern part, purple-colored carrot was usually grown. That's why I chose also Konya area. Total nucleic acid we did by stop protocol, and 1 gram of tissue from seedlings of each cultivar was used. Nested PCR, we did nested PCR by using the standard PCR primer pairs. followed by FU5 and RU3 primer pair. And PCR products were separated on agarose gel and stained by etidine byromid, and applicant size was detected and visualized under the UV imaging system of GeneGenius. Phylogenetic analyzing and sequencing, the Sanger method was performed by a commercial firm to reveal the bidirectional sequencing of total 16 PCR products from 4 wheat samples and seedlings. Blast analyzing of NCBI was used to verify the results with those other phytoplasmas in the GenBank. The sequences were submitted to NCBI database under the accession numbers MZ463052, MZ463020, and was used for alignment with other phytoplasma sequencing using cluster W in Mega10 software. In silico PCR and RFLP analysis, computer-stimulated PCR RFLP analysis were performed using the SnapGen to improve the accuracy of phytoplasma classification based on the partial 16SR RNA gene sequences. For this purpose, a total of 16 nucleotide sequences obtained from the carrot seedlings and weeds were digested in silico with RSA1 enzyme. Results: main clearing, reddening at the leaf tips, yellowing, and severe reddening and purpling were observed in one of the each Synapis arvensis, Conium maculatum, Medicago sativa, and Dacus carota white, respectively. There was also no phytoplasma symptoms in all seedlings at cotyledon stage. The main symptom of the infected carrot plants were reddening and chlorosis of the leaves. 6 out of 7 commercial seeds were all infected with phytoplasmas. Amplification were produced in nested PCR and expected 883 base pair applicants were obtained. Positive results were obtained from the most of the carrot seedlings and from 4 out of the 19 wheat samples tested. Now you see the general view of our carrot fields. In the carrot fields, you cannot see much weeds because the grower using herbicides before planting. Now you see the chlorosis and the reddening starting from the tips of the leaves. This is general view. And this is also samples of carrot leaves, reddening symptoms, and also chlorosis. It starts as chlorosis symptoms and then turns to be reddening. Yes, it's also chlorosis on the carrot leaves and then turns to be reddening, severe reddening. Those are the root symptoms of infected fields. There are many taproots also were seen, and distortion in the roots of the carrots or less growth. Those are the weeds, especially we found surrounding the carrot leaves, Conium maculatum. It is a poisonous weed, Fumaria officinalis, and Synapis arvensis. Candidatus phytoplasma tyrifoli infected conium maculatum. It's the reddening of the leaves, and also the Cuscarota wild. This is after we do research and we check the pictures. They were all infected with Candidatus phytoplasma tyrifoli 16SR6A subgroup. This is Candidatus phytoplasma tyrifoli infected Sinapis arvensis plant. There was a really sharp vein clearing symptom on this infected plant, and also phyllodes on Medicago sativa. This is ALU1 digestion of uncharacterized DNA isolates with 16 F2R2 primer. In this picture, you can see weeds and also plant samples, and also some insects, which I didn't include in my speech. This is 16 SR6 isolates. This is 16 SR1 isolates, yani Candidatus phytoplasma asteris. Candidatus phytoplasma asteris was B group, B subgroup, but Candidatus phytoplasma tyrifoli was A subgroup. This is ALU1 digestion of Konya isolates in the same procedure after amplification of F2R2 primer. Yes, this area is completely Candidatus phytoplasma tyrifoli, and also we have seed and the seedlings isolates here, and then Candidatus phytoplasma asteris. Candidatus phytoplasma asteris infected carrot leaves, and also the root system. It also, we also got very little roots because of the phytoplasma. In Konya, especially the purple-colored carrots were grown there. You know, a root system was so small, and we had a lot of lateral roots, as you see, and severe redness of the leaves. The sequence of CS8C strain obtained from the commercial carrot cultivar was 99% identical to Iranian strain of Candidatus phytoplasma asteris strain. The remaining strains showed 99 sequence identities with some Candidatus phytoplasma tyrifoli strains. In the phylogenetic tree constructed on 883 base pair fragment using the sequences different phytoplasma group or subgroups, the phytoplasmas from 4 weeds and 11 seedlings strains clusters with Candidatus phytoplasma tyrifoli. Only CS8C strain clustered with Candidatus phytoplasma asteris. After comparing the computer-stimulated RFLP analysis with RSA1 enzyme, 15 strains were enclosed in 16 SR6 and 16 SR1 groups. This is in silico digestion profile of carrot Candidatus phytoplasma tyrifoli and Candidatus phytoplasma asteris. This first one is with MSE1 enzyme, and the second one is with ALU1 enzyme. This is Candidatus phytoplasma tyrifoli, and this is Candidatus phytoplasma asteris. These are the phylogenetic analysis of our isolates. Mostly was 16SR1B, and we had no Stolber isolates. We were expecting, but we couldn't get any Stolber isolates. And also 16SR6 isolates here. We had lots of sequences in the gene bank from carrot also. So this area only shows our seedlings and also weeds. Group 6 here, 16SR6. This is A group, and 16SR1 group. This is Candidatus phytoplasma asteris. This is Candidatus phytoplasma trifoliae. It is the widely seen here. This study aimed to reveal the seed and weeds as an inoculum sources that play critical roles in the spread of phytoplasma diseases associated with reddening chlorosis and reduced root quality, widely observed in larger carrot growing areas in Turkey, Ankara, and Konya. Transmission of phytoplasmas through seeds was somewhat a controversial topic and has long been considered impossible due to the lack of direct connection between phloem system and embryos. But the system is working now. Transmission of phytoplasmas through seeds was reported in tomato corn, winter oil rape, as well as brassica napus. The presence of phytoplasmas on carrot seeds was first reported by Carminati, and it was confirmed by Satta at 2020, reporting that the seedlings were infected at cotyledon stage with one of the groups, 16SR1 and 16SR12. This present study confirms the presence of 16SR1 group and detect 16SR6 group in the seedlings of carrot cultivars for the first time. The two phytoplasma groups were not found together in any of the infected seedlings grown from the same cultivar. This present work also revealed that symptomatic Medicago sativa, Synapis Dacus carota wild, and Coni maculatum weeds were infected by phytoplasmas in 16SR6 group, phytoplasmas in 16SR9C and 16SR12A phytoplasma subgroups has been reported in Synapis arvensis, Conium maculatum, and Dacus carota wild, respectively, by Casati and then Satkaya. Therefore, according to our best knowledge, it was the first report of presence of 16SR6 group phytoplasma in this wild species, Synapis arvensis, Conium maculatum, and Dacus carota wild. This project was also founded by Bilecik Sheikh Adibali University Scientific Council, because my quota for Ankara University was already full, so the other university we got found. Thank you for your attention.
Thank you for your presentation. Unfortunately, we are a little bit late, so there is no time for questions. If you have questions, you can discuss during the coffee break. Thank you. And we are going to the next presentation.
Okay, we are a bit late, but we hope to meet at the lunch with some delay. The next presentation is seed transmission of phytoplasmas in tomato and chili varieties commonly grown in Mauritius, and it is given by Arti Gungu Singh Bunwari. Arti, thank you.
Thank you, Prof. Let me put the presentation. Okay. So, good afternoon, everybody. It's already noon. My name is Arti Gungu Singh Bunwari, and my presentation is on the seed transmission of phytoplasmas in chili and tomato varieties commonly grown in Mauritius. This study was undertaken in collaboration with Nicoletta and Professor Assunta. For those who are not very familiar with Mauritius, Mauritius is a small island in the Indian Ocean, only around 2,000 kilometers square, 900 kilometers east of Madagascar, and population is slightly less than 1.3 million. And the Republic of Mauritius is composed of a number of islands, among which you have Mauritius, the main island, then you have Rodrigue, Sagariga, and Sembrando. Mauritius is quite known as a touristic place. You have nice beaches, so you're all invited for holidays. But coming to the theme of the day, occurrence of phytoplasma-associated diseases in Mauritius on tomato was first reported in 1999, but at that time, not much details was given on the phytoplasmas. So as from 2002, when my collaboration with Professor Assunta started, we further investigated in the phytoplasma diseases affecting tomato. So then we got 3 phytoplasma groups. These were number 1, aster yellows. Then we had number 2, Stolber group, and number 3, Ems yellows group. It's quite difficult to distinguish between aster yellows and Stolber based on visual symptoms only, because both of them cause plant stunting, apical bushiness, and you also have some purple coloration. Although in the case of aster yellows, you tend to have more of the yellowing. For M yellows, it's a bit easier because you have shoot elongation and you have leaf distortions. But the problem is that at times you also have mixed infections of phytoplasmas or mixed virus phytoplasma infections, such as phytoplasma with potato virus Y or phytoplasma with tomato yellow leaf curl. Then it becomes more difficult, so there is a need to do molecular testing. Also in the past, we thought that phytoplasma transmission was being caused by insects mostly, because we noted some insects at field level. But then we did have some doubt that the disease was being transmitted by seeds also, because we noted phytoplasma diseases under strict insect-proof conditions, and we also found it in isolated places. So we decided to investigate more on the topic. We started a study with the sponsorship from the MRIC and together with University of Bologna. And the study was extended to Chile because we also noted some abnormal leaf distortions and yellowing on chili plants being grown in the vicinity of phytoplasma-infected tomato plantations. The major objectives of the study were firstly to investigate on the presence of phytoplasma diseases on chili, and secondly to verify phytoplasma seed transmission both in tomato and chili varieties commonly grown in Mauritius. So we did some field surveys and sampling, just like in the other presentations given. We covered 5 varieties of tomato. There was Swaraksha, MST32, Pêche-Rose, Topinas, and Francesca. In the case of chili, we covered 3 varieties, and we collected up to 5 symptomatic and 3 asymptomatic samples from plants. And all the plants where we did the sampling, they were tagged. So phytoplasma symptoms observed in chili plants were apical bushiness, stunting, leaf yellowing and distortions. The leaves were smaller, and we also have leaf and fruit distortions. In the case of tomato, again, we had the apical bushiness, the stunting, yellowing, and purple coloration. For testing, we used 1 gram of leaf midribs. We did the DNA extraction, and the DNA extract aliquots were forwarded to the University of Bologna, where they did the phytoplasma detection by nested PCR. Once we had the results, we went back to the field. We collected the mature fruits that had been tested positive. I mean, the mother plants had been tested positive. We extracted the seeds, we dried them, then we sowed them under insect-proof nurseries. Once we had the seedlings, leaves were collected again, and these were again, DNA extraction was done again, and these were tested again. Now, this slide shows the PCR schemes and primer combinations used. Coming to results, on tomato, the disease incidence at field level ranged from 5 to 60%. Highest incidence was recorded on one variety called Topinas that was in the south of Mauritius, and it's seen here. It was in a shade house, and we collected fruits, and quite surprisingly, this time we had only one phytoplasma group, that was the Aster yellows group. It was detected from 75% symptomatic mother plants from 3 varieties, that is Topinas, Swaraksha, and MST32. Germination rate in the seeds was 85%, and only 1% seedlings from only one variety tested positive to Aster yellows. In the case of chili, disease incidence was 10 to 50% at field level. Again, DNA extracts were dispatched to University of Bologna. 3 types, Aster yellows was identified on all 3 types of chili surveyed. Again, fruits, seeds extracted from fruits, they were sown, and this time we had 4.2% seedlings that showed Aster yellows positive. This slide shows the RFLP identification of phytoplasmas in chili and tomato seedlings that came from infected mother plants. As conclusion, we can say that the occurrence of phytoplasmas in chili is confirmed. Seed transmission of Aster yellows phytoplasma is confirmed in tomato variety Swaraksha and chili variety Pimosipai, so in only one variety of each. And then the symptomatic leaf samples that was tested negative to phytoplasmas, we had around 25% symptomatic leaf samples that were negative to phytoplasmas. This could be due to insect damage or due to the presence of viruses that I mentioned earlier. Before ending, I'd like to thank the management of Fairway, where I work, and my colleagues from the plant pathology department, the MRIC for sponsoring the project, Professor Asunta, Nicoletta, and not forgetting Samantha for the collaboration, the EU-funded project day2 for sponsoring my participation in this workshop. Before ending, I'd like to thank the organizers of IPWG 5. Special thanks to Professor Ali and his team for taking care of visa applications, for the warm welcome and the hospitality received here. Thank you. Thank you for your attention. So
thank you, Ati, for saving time. There is time for 1 or 2 questions.
Yes.
Because seed transmission is a very controversial issue, so I have a question about where you collected your infected mother plants, and then where you cultivate your new seedlings. Because I think the seedlings should be cultivated in the insect-free environment.
Yes.
Yeah.
So the fruits were collected from open field or from greenhouses, but once we had the seeds, they were sown in insect-proof greenhouses, and we placed some yellow sticky traps inside just to make sure there were no insects inside the environment where the seedlings were being raised. So they were insect-proof, so we can associate the symptoms and the presence of phytoplasmas to the seeds only.
Okay, some question from
You didn't get me.
Other question? There are questions from the web, no? Okay. Thank you, Arti, for your presentation.
Next presentation is seed transmission of phytoplasma infecting eggplants in India, and it will be given by Shri Tej Mateti.
Good afternoon, everyone. I see many faces are already tired, and I know we are late, so I'll try to finish as much as possible. Directly diving into the presentation. Oh, sorry. Okay. To give a bit of background, eggplant is an important vegetable crop and is considered as the king of the vegetable in Indian subcontinent. And people from India know that we call it as brinjal. And the production of eggplant is affected by many diseases, and one among them is the little leaf of brinjal, which is a phytoplasma-associated disease that is widespread in India. This disease was first reported way back in 1938, and the infected plants usually show symptoms such as reduction in the stem internodes, the presence of the tiny and small leaves, the axillary bud enlarged, but the petiole and the leaf stays short, giving it a bushy appearance, and eventually followed by the presence of the phyllodes. And due to the alternations in the floral structures, the plant bear no fruits or the fruiting is rare, and this is the reason why sometimes the yield loss is up to 100% and is a concern to the agricultural industry in India. The phytoplasmas responsible for disease spread through the insect vectors. However, as we all know, the seed transmission is also possible. But the research on data on the seed transmission of phytoplasma is limited. Therefore, this study was aimed to verify the seed transmission of the phytoplasmas from infected mother plants to their progeny. So far, phytoplasmas enclosed in 6 ribosomal groups have been identified to infect eggplants worldwide. Group 1 was identified in India, Bangladesh, and in Japan. Group 2 was identified in Egypt and in Oman. And I have to mention the seed transmission of phytoplasmas was first identified in alfalfa in Oman. Coming back to the presentation, Group 3 was detected in Brazil, Group 6 was detected in Turkey, and Group 9 was detected in Iran, and Group 12 was detected in Russia. That being said, multiple groups were also identified in the same country. For example, speaking about India, different phytoplasma groups were identified in majority states of the India. But the seed transmission of phytoplasma in eggplants was not reported yet. Our research starts from here. Symptomatic eggplants referable to the presence of the phytoplasmas was found in the Garga village and in short located in the southern part of the India. The eggplant mother plants were grown in open field conditions, and seeds from 2 different fruits belonging to the same mother plants were collected in May 2021, and later in August the seeds were kept in germination chamber for 3 days, and among the germinated seeds, selected number of seeds were transferred into the potting mixture in an insect-proof greenhouse. Among the 516 seeds collected, 471 seeds were germinated. The remaining seeds were either non-germinated or rotten. So as the seed number was high, we divided the seeds into 4 batches for 1 fruit, and even the germination percentage of all the batches was almost high. So we transplanted only 30 germinated seeds per batch in the greenhouse. And again, the survival rate after transplantation was also high in almost all the batches. And even the average percentage of germination and survival rate was approximately 91%, which is in line with the previous results on the seed transmission of phytoplasmas in other crops, for example, tomato. The plants grew without any symptoms except a bit of leaf yellowing, and even some plants produced fruits, and the fruits were also very normal. But when we dissected the fruit, we observed that some seeds are already pre-germinated inside the fruit, so it is decided to test even them, which I explain in the upcoming part of the presentation. And moving on to the molecular analysis part, the DNA extraction method we followed is the CTAB. As again the plant number was very high, DNA extractions was performed on selected plant samples based on their growth at different time intervals, such as 50, 60, 70, and 76 days after transplanting. For the identification of the phytoplasmas, amplification of 16S ribosomal RNA gene was carried out using different primers in 2 PCR schemes. And for the characterization of the detected phytoplasmas, amplification of non-ribosomal genes such as isoleucine and SEK gene was carried out using respective primer pair in the first and second rounds of the PCR. At 50 days time interval, few plants tested positive in nested PCR, and the RFLP profile of the samples were identified to be phytoplasmas belonging to group 12 and group 1. For 2 samples, the RFLP profile was not clear, and this is further confirmed by the sequencing analysis, in which a quantity of 802 nucleotide size 99.63% identity candidatus phytoplasmas solani for the group 3 positive tested amplicon, and that for of the group 1 phytoplasma shows 98.83% identity to candidatus phytoplasma asteris. At 60 days time interval, 3 more plants tested positive, and the RFLP profiles of one sample were identified to be phytoplasmas belonging to group 2, and that of other was group 6, and the other was a group 5. At 70 days time interval, few more plants tested positive, and this time the RFLP profiles of all the samples were identified to be phytoplasmas belonging to group 2. In particular, the profiles were exactly matching to the reference strain of subgroup B, and this is further confirmed by the sequencing analysis, in which a quantity of 994 nucleotides has 99% identity to candidatus phytoplasma arentifolia. And we also tested few more plants at 76 days time interval, but none of the plants tested positive. As I mentioned earlier, when we dissected the fruit, we found that seeds were already germinated inside the fruit, but since not all the fruits are germinated, we divided the seeds into 3 categories. One, the only seed, the another one is the germinated seed, the another category is the hypocotyl, that is totally cut from the seed, and we extracted the DNA again from all the fruits, and among the 16 samples, 8 samples tested positive in nested PCRs, and the RFLP profiles of 5 samples were identified to be phytoplasmas belonging to group 1, and that of other 2 samples were identified to be group 12. However, for 1 sample, the RFLP profile was again not clear. In addition to what we did so far, eggplant samples which are growing in the same field but in the next growing season were also collected, and the molecular analysis was carried out using the similar protocol, and all the plant samples were positive in the nested PCRs, and the RFLP profile of 1 samples were identified to be group 3, and that of other 2 samples were identified to be group 6. However, for few samples, the RFLP profiles was not clear, and again through the sequencing analysis, a counting of 840 nucleotides has 99.76% identity to candidatus phytoplasma pruni for the group 3 positive tested amplicon, and that of group 6 phytoplasma shows 99.80% to candidatus phytoplasma trifoli. And for the characterization of the detected phytoplasma so far, amplification of 9 non-ribosomal genes was carried out on all the positive tested phytoplasma ribosomal groups. However, only 1 sample, that is the field collected sample, was positive for both the genes, and this is again confirmed by the sequencing, in which the quantity shows 100% and 99.76 to 100% identity to candidatus phytoplasma trifoli strains of little leaf of brinjal from southern India for both the genes. To summarize the entire research story so far, at 50 days time interval, group 1 and group 12 were detected. At 60 days time interval, group 2, group 5 and group 6 were detected. At 70 days time interval, group 2 was detected, and at 76 days time interval, none of the plants tested positive, and in second generation seeds, group 1 and group 12 were detected. Moving on to the final part of the presentation, the presence of a germinated seed positive for phytoplasmas in fruits grown under the greenhouse is a proof that the phytoplasmas are transmitting through the seeds. And even among the 5 phytoplasma ribosomal groups detected during the research, group 1 and group 12 were identified both in the first generation plants and in the progeny, Particularly, the group 1 phytoplasmas was identified in the first generation and in the progeny belonging to the same plant, which is a significant confirmation that the phytoplasma is transmitting through the seeds. And group 2, group 5, and group 6 were detected only in the first generation plants. And interestingly, the plant tested positive for group 2 phytoplasma in first generation. Its progeny tested positive for group 12 phytoplasma. And moving on to the field collected samples, group 3 and group 6 phytoplasmas were detected, which indicating that the transmission is happening through the insect vectors in the same field. And our research results support the literature that the phytoplasmas were detected most abundantly in the early growth stages of the crop and are often different from those identified in the later stages. Moving on to conclude, the research on the seed transmission of phytoplasmas is important, as the seeds from the infected plants can affect both the quality and quantity of the production. The overall seed transmission rate is low, but the problem is the seeds are helping to disseminate the pathogen in the new areas in the presence of the insect vectors. Therefore, more research is needed to understand and to acquire proper knowledge on the seed transmission of phytoplasmas in eggplants or in any other crop for that matter, to plan better management strategies and to reduce the impact of this disease on the crop. And lastly, the research alone is not enough. In fact, the collaboration between the researchers, farmers, and policymakers is crucial to address this issue. Here are some research questions. Hopefully, they will be addressed in the future. And I would like to end up my presentation with a quote said by Albert Einstein: "Keep on sowing your seed. For you will never know which will grow. Perhaps it all will." And thank you all for your attention.
Thank you. Are there any questions? If not, do we have some online questions? Okay, if not, maybe I can have one. How you think it comes that by testing seeds from India you get, I think, Stolbur, and if I remember well, group 3 phytoplasma. Do you hear me? Okay. Which are not in general present in the area where the seeds come from.
To answer your question, we are also wondered the same. The group 3 was never detected, especially in eggplants, but we found it, and which is again further confirmed by the sequencing. So we have the group 3 phytoplasma. Yes.
Okay, thank you.
Thank you all for your attention. I hope I don't took much time. Bye.
Yeah. So our session is finished now, and I think we can go for lunch. Huh?
Okay. Since we do not have a lead, we wait everybody back at 2:30 after lunch. Of course. Remember, the poster are across to look at the poster. Ali is here.
Yeah, now ready for lunch? Let's go. Same place where it last night, yesterday. And we'll come here at 2 o'clock, 2:30. We'll meet here again at 2:30.
So I think we should open now this afternoon session. Please take your seats so that we will not be too late this afternoon. We have the dinner tonight. And we will talk about the control of phytoplasma-associated diseases in this session, in this afternoon session. I invite Attilio Rizzoli to present adaptive management trials for the control of Scapuideus titanus, main vector of Flavescens doré phytoplasmas. Please.
Thanks a lot. Good afternoon, everybody. I would like to thank my colleague Christophe de Bonneville, also Fagroscope, and his boss Olivier Schump. And then I'd like to also thank my colleagues at WSL, Swiss Federal Institute for Forest, Snow and Landscape Research, and the Plant Protection Service of Canton Ticino. So, I won't bore you on the introduction, since we already had colleagues talking about FD. I just want to show you this nice picture of Chardonnay, showing extreme expression of grapevine yellow symptoms. This comes from the last vineyard that we had to grow completely in Canton Ticino in the south of Switzerland, where we had 20% of incidence. And this is not the result of having issues with the control of Scaphoideus titanus. This is the result of negligence, of human negligence. So for the people who are not from Europe, just a small reminder of where our tiny country, Switzerland, is located. It's located in the middle of Europe, on the European Alps. And regarding Scaphoideus titanus, we have two major areas which are colonized by the vector at the moment. We have the Southern Alps, Canton Ticino, where Scaphoideus is present since the 60s, and then we have the western part of the country, which will be discussed more in depth by my colleague. As for Flavescens doré in planta, we had the first detection in 2004 in Canton Ticino, and the situation got out of hand quite quickly, so we are now dealing with FD as an endemic disease in Canton Ticino. That's why the solid red area is all labeled as infested, and we have a buffer zone where we also apply control measures to contain the spread, while on the western part we have single infested zones. And as I said, Christoph will talk about them later on. I will focus then on the Canton Ticino, the southern part of the country where I'm from. A little bit of chronology regarding Scaphoideus titanus and Flavescens doré up to 2018. As I said, the first observation of Scaphoideus titanus was done in 1967, absolutely by chance. Then decades went by without having any issues. In the 90s, my group leader started working on the biology and ecology of Scaphoideus titanus in southern Switzerland, just to get ready for the arrival of Flavescens doré, which arrived, or it was detected at least for the first time in 2004, always in Ticino, because we have everything in Ticino, of course, being next to Italy, on the cultivar Gamare. And so in 2005, the mandatory insecticide applications and all the other usual classic control measures were implemented, starting from the municipalities where Flavescens doré was detected in planta. And we used buprofezin, which is still known with its commercial name Applaud. Then from 2006 to 2018, FD kept on spreading, and it arrived to cover almost the complete wine growing area of Canton Ticino. So the problem was that we used an effective active ingredient, which is known to be quite good in the control of Scaphoideus titanus, but Flavescens kept on spreading. We had, of course, issues also with the wine growers who didn't understand what was going on and why, as researchers and as extension services, we couldn't help them find the solutions for the containment of the disease. And we also had other considerations, that buprofezin in general worked quite well. The vineyards that were surveyed by the colleagues were indeed not that much abundant in Scaphoideus titanus. But of course, this kind of mandatory treatments have a high economic and ecological cost. Moreover, 85% of the wine growing surface in Canton Ticino is cultivated with Merlot, which is known to be less susceptible to Flavescens doré. And as I said, Flavescens kept on spreading regardless of all these considerations. So the idea came up to think about new strategies, and the first came up with if we stop applying insecticides for 1 year or 2 years, what happens? which is the remount of the Scaphoideus titanus population inside these vineyards. So with agroscope, we started a preliminary study in 2016 in 3 Merlot vineyards. And just to go a little bit quick, in the following 2 years, we did not find a significant increase in Scaphoideus titanus populations. It's important to note that the initial populations in 2016 were nearing 0. Then, in 2018, we also had more like a political problem, so to say, or issue. The authorization for the use of buprofezin was retired. And this was quite an issue. And we then had the authorization all just for pivefrins. And we asked as an emergency authorization spirotetramat, which is also known as Movento. This was, of course, a further push in the direction of the need to develop new strategies for the control of Scaphoideus titanus. So after this pilot study by agroscope, we came up with the colleagues of WSL and Canton Ticino to propose a wider project regarding the adaptive management of Scaphoideus titanus in southern Switzerland. And the Federal Office of Agriculture accorded a suspension of 2 years. So even though we are in a situation where the insecticide applications are mandatory, we received, regardless, the possibility to suspend the insecticide application for 2 years. So the area, as I said, is in Canton Ticino, just to give you a little bit of an idea of where we are from. And the collection of data was done during the period 2018, profiting from the last year of the pilot phase of agroscope, and that lasted until 2022. We worked on 9 vineyards, 7 cultivated with Merlot and 2 with Chardonnay, which is known, the opposite to Merlot, to be quite susceptible. The monitoring of the vectors, not just Scaphoideus, but also the alternative vectors such as Orientus Ishide, was conducted with yellow sticky traps, Rebel Giallo, and we had at least 6 sticky traps per vineyard, changed weekly from June to October. Of course, all symptomatic grapevines were sampled and sent to the lab for molecular analysis. The results are just coming from the 2 figures that I put in the abstract. I'll try to go step by step. So we had the initial situation in 2018 where we had buprofezin, and we had this situation. So populations, this is the mean captures per trap per year. So it's like 2.3, 2.02 something, which is nearing 0 if you consider it over the whole season. Then we started the suspension regime, and of course, as expected, we had an increase in populations. We had the second year with further populations increase, as expected. But if you just saw the numbers, at least the mean values, you'll notice that, yes, the rate is quite high, but we are not reaching crazy numbers per trap per year. Then we had to reapply pivefrins to see the efficacy of a treatment based on this kind of active ingredients, with the strategy of using 2 sprayings 14 days apart post-flowering. So we also, as other colleagues, try to place the first treatment on the peak of the 3rd nymphal instar, but we also have to consider the phenology of grapevines, which is also something we have to follow according to regulations in Switzerland. And this is what happened. We had a decrease, as expected, but a decrease which is absolutely not enough to contain a population or to come back to the safe zone, so to say. Second year, and we reached sort of a flat curve. So the second year did not manage to further decrease the populations. It just contained the remount of the population for 1 year to the next. So yes, in the end, we have still 1.5 times the population of suspension year 1. So we have a positive increase over the years. And if we compare all the groups, we don't have any significant difference when we consider all the plots. This is a little bit misleading, maybe, if you watch the figure, but if you looked up left, sorry, it's standard error and not standard deviation, just to be sure about it. So it's really not significant. What's important, of course, it's what happens in vineyards. So the incidence of infected grapevines is what actually matters to us and to the wine growers. And this is what we saw. We had absolutely no significant differences in the incidence of infected plants. And as you can see, we serve it from around 10,000 to 24,000 plants. So we are not talking about small plots of 200 or 400 plants. And we had absolutely no significant difference. This is probably also due to the fact that Merlot helped us, but also Chardonnay did not have significant increases in the incidence. So, of course, are insecticides then useless or unnecessary? Absolutely not. Absolutely not. But it's obvious that in terms of cost benefits, something must be reassessed. It's not viable to say just we have to use insecticides and then just let it be to the wine growers to just do the treatments without being guided or aided in any way. The high variability is also explained by the quality of the application, because we have wine growers working really well, and we have wine growers not following the regulations, not following also what we try to tell them to help them. And it's important to consider that in the specific case of Canton Ticino, southern Switzerland, we had buprofezin, which was really effective in the control of the populations. So we had several years of continuous mandatory applications, which led us to have a low initial population before the suspension. Of course, quite important, we have no abandoned vineyards in the proximity of cultivated plots, which is a problem in other regions. And the systematic roguing of infected grapevines was really kept as a must. Nothing was left in the field. And as I said, and I will say it once more, we are lucky that we have 85% of the wine growing surface cultivated with Merlot. Of course, when we talk about insecticides and active ingredients, we always want to talk about efficacy. But efficacy does not depend just on the active ingredient. It depends on the quality of the treatment, on the quality of the application of such active ingredients. And I'm talking about spraying method, the removal of suckers, which is really important, especially in the case of Scaphoideus titanus, and the vegetative wall management, which is especially important for the treatment with pyrethrin. In fact, we observed also in other experimental vineyards of the area efficacy levels ranging from 20 to 80%. So pyrethrin are still doing a good job when properly applied. The problem with pyrethrin is also that we have strong regulations in terms of distance, the distances we have to keep from surface water. So we can't use them if we are near to surface water. We have to be careful with bees. We have to treat in the morning, early in the morning or late at night, because UV can deteriorate the pyrethrin. So it's not an active ingredient very easy to apply, especially on bigger surfaces and especially for the professionals who have many hectares of vineyards scattered around. And of course, as we all know, there's a high political and social pressure on the use of phytosanitary products. And another big issue involving also the industry is that there is a big uncertainty on the longevity of active ingredients. So there is no push for development of new active ingredients because we don't know if the authorities will allow for the use of such active ingredients. And in the case of pyrethrin or acetamiprid or other active ingredients, if we use them alone or in a consecutive year, we can also incur in the emergence of resistances. So to conclude, adaptive management is indeed achievable. It it is achievable. And I think that if you consider special conditions such as those we have in southern Switzerland, we could maybe try once again to apply it. If we can have low initial vector populations, so nearing zero, of course. And of course, we need a perfect implementation of the accompanying control measures, so removal of all infected grapevines, the use of certified planting material, hot water treatment, etc., etc. And absolutely important, it's the eradication of abandoned vineyards and absolutely eradication of gone wild grapevines, which is something that also in southern Switzerland is becoming an issue that I wouldn't say we weren't aware of, but if you look, if you search, you're gonna find them. If you don't look for them, you're not gonna find them. So just be aware of them. And of course, the use of, or the use of adaptive management is a strategy, especially in situations where we have suitable cultivars. So for example, also in the case of Tokai friulano, it may be interesting to apply such measures and not just think about the potential of recovery of plants when already infected by phytoplasma. And I would say the key issues are the spraying application. Of course, it depends on the machinery used by the wine growers, the timing of the application, and good agronomic practices in general. And here is also something that I would like to discuss with you colleagues, the availability of effective active ingredients, because pyrethrin are doing okay in certain situations, but the problem is that it's going to be more difficult to achieve a situation of having low initial vector populations before reapplying a possible suspension of treatments. Thanks a lot.
Thank you for this presentation. Are there questions? No questions?
Sorry, Attilio. I have just a question. Because I completely agree with your observations. Just what about the percentage of the grape growers that don't apply insecticide in your country? Because I think that if just one or the minimum percentage that not apply insecticide can affect the results of the entire grape growing area.
I absolutely agree on your consideration. The special case of southern Switzerland is that we don't have a continuum in the vineyard area. We have many plots scattered around. And usually when we have a continuum, we also have a stronger relationship between the different wine growers. And there's also social pressure, I would say. And usually they also work together to plan the treatments. So it's more a problem with hobbyists, of course, and with private gardens. That's where the problem for me is.
Thank you.
Yeah, I had a question. The measures which you suggest, there are obviously going to be more pesticides being withdrawn from the market. Do you think there are sufficient? Are there other options you're investigating also?
I must say I'm not that confident. I'm not a big fan of the use of pyrethrin for consecutive years. I think that many of us know that there are many negative effects on the consecutive use of pyrethrin. At the moment, for southern Switzerland, we also got the chance to have this transitory authorization for spirotetramat, which may be a solution to just get milder effects on the beneficial organisms when using pyrethrin. And when it comes to active ingredients in general, I don't see a future for them, especially in viticulture and especially in Switzerland, because the pressure is really high.
Just a clarification. You mentioned that the FDA arrived in Switzerland because you border Italy. but we have mountain in between. I think it is more wise to, no, we have continuous in some place. I know, but there is no viticultural area there. Maybe there is some vineyard. It's not so intensive viticulture. So I think you should also consider the propagation material as source.
Of course, I was joking. We don't. No. You're right. You're right. That's important. It's not. It was a funny joke because with Italians and Swiss in the southern part, they're always funny with each other. But it, of course, could be the use of uncertified propagation material that caused the first detection. And we are almost sure that 2004 is just the year that the detection was was made. But most probably it dates back years. Also because this this gamma ray plot had an incidence of 18%, if I remember correctly. So not something to play around. Sorry for the.
So do we have guests online? Yes, I see there are guests online with questions.
I think it's me. Hi, my name is Esmeraldina. I would like to ask if, I don't know if I can to speak.
Yes,
are
we can hear you,
hearing
please.
me? Can I speak?
Yes, we hear you. You can continue.
Okay, thank you. I would like to know if you have the putative vegetable host as alder and ailanthus in the border of the vineyards you study. Because I think I agree with you, we need an adaptive management to control Flavia Sansore, but it depends a lot of variety, and you have the Merlot and the agriculture landscape. In Portugal, it's very difficult to control, to not do the treatments, because we have smart vineyards and very near each other, and we have the putative, the vegetable host, and we have scaphoideals and other putative vectors also.
I absolutely agree. Of course, I didn't have the time to mention all the rest of the work that we are conducting at Agroscope and WSL. Actually, I'm used to talk about the role of alternative vectors and those plant. So it's quite funny that I'm just talking about scaphoideals, but it's the biggest theme, the biggest topic of our research is the role of the wild compartment. That's why I also said something about the gone wild grapevines, because they are obviously a host of Scaphoideus titanus, and not just Scaphoideus titanus, I would say, but we are a meeting of phytoplasma, so I guess you can make 1 + 1. And regarding alder, yes, we already did, we wrote a paper in 2019 relating the FD genotypes found in alder and Orientus Ishide, which we made also in collaboration with Elisa Angelini, who is probably watching us. And we also wrote an article on the role of the putative vectors Ischimonus amatus, Graphocephala fennai, and Japananus hyalinus. Just tell you that we are indeed also considering the possible role of alternative and putative vectors and host plant species. But in general, even if it's really, really difficult, and I wouldn't believe anyone who would say otherwise, I think it's really difficult to quantify the role of these secondary or alternative epidemiological cycles for now. So I don't see how we could integrate this at the moment, for example, with a model. We usually have M54 in vineyards. We do have instances of M50, we already had M38, but usually almost 100% of the observations is M54, so we know that it's spread by Scaphoideus, and it's a thing that it's linked to grapevine. Thank you.
Okay, thank you.
Okay, many thanks.
Thank you.
Thank you, Attilio. We can move with the detailed assessment of control measures against Flavescens doré allows a reduction of pesticide use. The speech will be given by Christopher de Bondeville.
Thank you. Good afternoon, everybody. I hope you still have some energy to get a little bit more of the Flavescens doré team. So I'm going to present a part of our work.
More close to the microphone.
Like that's better? Oh yeah. I'm going to present part of our work which is more applied to the practical control of the disease in the canton of Vaud. This work has been realized in cooperation with Agroscope and the phytosanitary services of the canton. On the left, you have a picture of the Lavaux vineyard in the Lake Geneva region. This is the region where we are, and yeah, if you're in the region of Geneva, it's definitely worth taking a look at this vineyard. It's several centuries old vineyard with a beautiful view on the Alps, on the lake, and it has to be all the work there is done in most of the surfaces by hand because the terrain is too steep to go with machineries. Okay, you probably noticed that I was not a tourist guide in a previous life. This is just a summary of the surface of the vineyards in Switzerland. As you can see, we have about 15,000 hectares of vineyards in the country, which is like a small drop in the sea if we compare to the big European and also American, South American producers. And our region is in western Switzerland, so canton of Vaud, which represents about 30% of the surface of the total Swiss vineyard, so a little bit less than 5,000 hectares. Oops, sorry. I won't go into the details. You've seen that many times. Maybe the most important points is the last points. For the FD detection in Switzerland, in north of the Alps. It happened 11 years after the first detection in the canton Ticino, which you just heard about it. And that was done in 2015. And it was introduced most likely by planting material. These are the control measures that are used in Switzerland for the FD disease. So there is a yearly prospection in vineyards as part of territory surveillance, and also yearly monitoring of the Scaphoideus populations. Of course, there is a mandatory announcement of any grape 1 yellow symptomatic vines, and a recommended planting of healthy material. So any planting material must have a phytosanitary passport. And there is, for the moment, only recommended hot water treatment of the planting material. We are trying to push to make it also a mandatory measure, the hot water treatment. In case of a confirmed FD positive stocks, there is insecticide treatments in a defined control zone and uprooting of all symptomatic stocks. Okay, Attilio already talked about the distribution of the Scaphoideus titanus. So you see that around the Lake Geneva, the insect is well installed. This is also the map of the FD disease affected areas. So Ticino is an endemic zone of the disease. In the west, you can see red points, and these are specific punctual outbreaks that appeared since 2015. In the specific region of Canton Vaud, we have several outbreaks in different regions, and they have also different severity when they were discovered since 2015. So the most severe ones are in the historical region of Lavaux, And also there is a big one completely east of the lake in the region Chablais. I'm going to show you a couple of examples from 2 outbreaks in the region of Lavaux. So what is being done currently in the canton is an extensive monitoring of the insect populations and also the symptomatic grain points in the controlled perimeters, and the risk assessment of each these perimeters. After the outbreak discovery, there is always 2 consecutive years of mandatory insecticide treatments. But from the 3rd year, based on this risk assessment, there is a decision if the insecticide treatment is going to be applied or not in the perimeter. So the treatment can be suspended if the pressure is low. And by pressure we mean both vector population and the number of infected gray points. So the first example is an outbreak that started in 2017, sorry, in the region of Chardon. So you can see on the right a table showing an estimated number of uprooted vines in the perimeter. So quite strongly affected. About 700 vines were uprooted in 2017 and 18, and a continuous decrease of the number of infected grape wines through the year until 2022. This number has not yet reached 0. We still hope it will one day. On the left-hand side, you have the red bars. The red bars, they're the samples that were tested and confirmed by laboratory analysis, molecular analysis in the laboratory. So of course, in the first year, with this great number, we did not test all of the samples. Some of them were only visually disease determined. And you can see also that. You can see also the curve of the insect population, which is in black. And the insect population was assessed by putting 20 yellow sticky trap in the perimeter during the peak of flight of the insects. And as you can see, after treatment with buprofezin, the population of the insect is very low. Here on the right, I don't know if you see the scale, but it's less than 0.5 insects per week and per trap. So very low population, as Attilio showed before. The ban of buprofezin and the move to pyrethrin did not significantly change this population decrease. So here there was a constant treatment with insecticide because the risk was still too high to stop the treatment. Here I show you another example, which is an outbreak which is a few kilometers away from the other one. And it is the first outbreak of the Canton Vaud, which started in 2015 with quite about the same number of infected plants than the previous one. And you see also the reduction of the number of infected plants with the measure applied in the canton. Here you have 3 consecutive years of treatment with buprofezin, which reduced the population of the insect almost to 0. And then in 2019 and 2020, there was a couple of treatment with pyrethrins, which did not do anything significant. And then it was decided in 2021, it was asked by the canton authorities to suspend the treatment based on the very low insect population. We are here also around 0.5 insects per trap and per week. And very low number of plants, less than 5 plants in the infected plants in the perimeter. It was asked to have an authorization to suspend the treatment of insecticide. As you can see, this had no effect on in the 2 first years of suspension on the insect population. Okay, so what's the status for this year? There are 8 different affected perimeters in the canton of Vaud, which is covering about 650 hectares. And for the next year, so for this year, there will be 200 hectares who will receive the 2 insecticide treatments, and 450 hectares which will be free of insecticide treatment. So what are the conclusions of this assessment? So we had a strong political and consumer pressure to push us toward a reduction of pesticide use. And we used a detailed assessment of vector populations and infected grape vines in every perimeter to allow to move towards this goal. The advantages of this strategy is a significant cost reduction. The insecticides are really, really expensive, and also a reduced environmental impact and an improved public image. The difficulty, and this is really a time-consuming strategy, and there is high need in human workforce, meaning that not only the authorities can do it, we need the help of all the wine growers from the region, which is not always evident to motivate to have. And we are also aware that 650 hectares is like a drop in the sea compared to the numbers you presented for only Veneto this morning. It's like 1% of the surface. So this type of strategy is only adaptable to the small scale of the vineyards in Switzerland. And with that, I would like to thank you all for your attention.
Many thanks, Christoph. Are there any questions? Wolfgang?
Thank you for the presentation. Just a question to your introduction. You mentioned that it's the announcement of grapevine yellow symptoms is mandatory, and then the symptomatic stocks have to be uprooted. My question is, don't you have Bois Noir in your region? Bois Noir, which you don't have to uproot probably.
Yes, we do have Bois Noir everywhere in Switzerland. So they have to announce any grape wine yellow symptoms, and then the only test in the laboratory would determine if it is Flavescens doré or Bois Noir phytoplasma.
You can do this for all the stocks which are announced?
Yes, it's not that much.
Okay. Online question, yes? Please?
Any question?
No, please, there is a question for web.
What?
There is one question from web.
Okay.
Please. No question?
No.
Here one question.
No question. Okay.
Maybe I have a final short question. So far we don't yet have Flavescens doré in Germany, and neither Scaphoideus titanus, but when I saw your strategy, even if it's low populations and it's not so, for example, for the Geneva region, not so widespread, only those spots, isn't it a bit risky, this strategy? Because then, after 2 years, you are able or allowed to suspend, and if there is a new outbreak, you have to restart again?
It is risky, I have to say, but this is why there is a continuous monitoring of the incidence and of the populations, and if it starts again, you have to do benefits and do the balance between the benefits and the risk, and it could be risky. I agree.
Okay. Okay.
Thank you.
Thank you, Christoph.
So we go ahead with the next talk, which will be given by Nicoletta Contaldo, Plasma Activated Water and Phytoplasma Interactions in Catharanthus roseus Alkaloid Pathway.
Good afternoon to everyone. I'm really happy to give this talk because this is I'm going to present some of the results obtained on the use of plasma activated water in infected micropropagated periwinkle shoots, and this was carried out. This work was carried out in collaboration with the group of the engineer at the Bologna University, led by Professor Vittorio Colombo, and carried out by Romolo Laurita, because this is a very fruitful, nice collaboration that we have with this group for the last 10 years. And first of all, what is plasma? Plasma is the fourth state of the matter, is the matter of the stars, they said. And everyone already know the plasma because it's the matter of the thunderbird. So like in the storm, like today, when you see the thunderbird on the sky, this is a plasma. And plasma, the flux of plasma can activate the species, the nitrogen, oxygen species in the air, so it produce the reactive ROS and RNS species. And this plasma has been used for several years, almost 100 years, maybe more, for different purposes. For the industrial, for example, for the industrial metal purposes, and or for the TV, or in terms of medicine, for example, in the surgery, to disinfect the surfaces or the instruments, and also for, I don't know, the sterilization of the wounds on the skins. But in our case, we use this plasma to produce a solution. So I produce this water called PAV, so plasma activated water, that is a special solution that contains these reactive species of nitrogen and oxygens that are produced by plasma generator. In this particular work, we use as a generator a DBD, so the electric barrier discharge, that was used to generate an on-equilibrium plasma in contact with a liquid surface. So the ionized water was treated with a nanosecond pulse high voltage generator in a closed chamber, and the volume of the reactor was filled on an ambient air and used as plasma gas. So we obtained, the engineer obtained this solution that is a cold plasma, so at room temperature, and with different species like nitrites, nitrates, and peroxides that are in different amount, and they have the different decaying time. The first, I don't want to go deeply in the chemistry of the PAV because it's not my field. But at room temperature, the concentration of hydrogen peroxide and nitrates start to decrease significantly after 2 days, and if you store at -20, after 3 weeks. So we use this plasma activated water for our purposes. Why? Because in the recent year, there is this new field, new rapidly emerging field, that is plasma agriculture. It was thought that the plasma activated water can be the future, because it's like just water, and so can be used in eco-friendly agriculture. And first of all, was used for the disinfection of the seeds, because it doesn't affect the germination. And then was used, for example, in the post-harvest with strawberry for the disinfection of the surface and against some fungi like Botrytis cinerea. And then we start to think about the use of this plasma activated water directly on plants. The first thought was it can be the renewable fertilizer, because it is well known that is help and enhance the plant growth and also the plant production. In our case, so when we start to collaborate with this engineer group, we, of course, want to explore its use in plant pathology. So we use the PAV treatment in different pathosystems, and what we found that this PAV enhance the plant's growth, yes, but induce also the plant resistance in different pathosystems, like when tomato affected with bacterial leaf spot, and in grapevine and periwinkle plants infected by phytoplasmas. And in particular, this plasma-activated water was found to trigger the expression of genes involved in the alkaloid biosynthesis. So this was the starting point for this work, because we know that the synthesis of alkaloids can be influenced by diseases or stress factor. So we want to explore the interaction of this plasma-activated water and phytoplasmas in the alkaloid pathway. Because we had the collection at the University of Bologna. There is the collection of micropropagated periwinkle shoots infected by different strains of phytoplasmas that is kept by Professor Bertoncini for almost 30 years. So we have the chance to have a lot of shoots infected with phytoplasmas. We have the plasma-activated water, so we want to explore the interaction between this water and the phytoplasmas in the alkaloid pathway. So, the experimental design, we use 100 Asteriellos infected periwinkle micropropagated shoots and healthy one, and we performed the treatment with different solution. So PAV, sterile distilled water, as a negative control, and phosphatidylene aluminium as a positive control, because it's a well-known resistant inducer used in the field. And we did the gene expression analysis on 5 genes in the phenylpropanoid pathway at 5 different time points: 0, 7, 24, 48, 96, and under 20 hours. And this is just the fact of the visible effect of this treatment on the shoots. In the middle, there is the periwinkle shoots healthy, and on the left, on the right, they are infected by phytoplasmas. But on the left, they were treated by PAV, and on the right, they were treated just with sterile water. So after 2 weeks of treatment, you see the morphology and the symptomatology of the plants are quite different from where they were before the treatment. So the leaves are larger. So they seems like with the reduced symptoms. Not the reduced phytoplasma presence, because the amount of phytoplasma, at least after 2 weeks, was the same, but the symptomatology was different. So the PAV has an effect also on symptoms appearance. But our purpose wasn't the control of phytoplasma at this time, just the effect on the alkaloid pathway. So let's check the different genes that we selected. The first one is the Qaboos synthase, and the 2 graphs on the left, there is a graph related to the infected shoots, and on the right, on the healthy shoots. A different time point with the different solutions. And with the arrow, you see the plasma-activated water effect. So that is in green in this graph. So the behavior of infected and healthy shoots are the same, and the PAV treatment enhanced these genes after 7 hours, so the time point 2, but not so high in comparison with the phosphatyl aluminium, that is the one in blue. So the phosphatyl aluminium in this case was more effective in comparison with PAV. And when we move to the phenylpropanoid pathway, the PAV enhanced very well the different genes in comparison with the phosphatyl aluminium and the negative control. In this case, we have PAV in purple, so with the 2 arrows. Again, the behavior of the plants, the infected and healthy, or the shoots, sorry, the infected and healthy shoots is the same, but we could detect an overexpression of these genes, the phenylalanine ammonaliase, after PAV treatment, after 24 hours in this case. And when we move through the pathway, and this is a streptocidin beta glucosidase gene, so start to change also the behavior of the shoots. Because on the left, we have the infected one, on the right, the healthy, and you see with the arrow, again, the PAV action. And with the infected plants, we have an overexpression at 2 time points after 48 and after 96 hours, while in the healthy shoots, the effect of PAV is more stable. So after 24 hours, it could enhance the expression of this gene. And at the end of the pathway, of the phenylpropanoid pathway, there is this gene, the acetylvindoline or acetyltransferase, that gene. And you see, we got this quite nice difference, because in the infected shoots, the PAV was active after 24 hours, and while in the healthy one was active only after under 20 hours, but not so in such a big way like in affected. So this was for us was like a key of this work. So in this case, with this gene, PAV and phytoplasma are affecting the same pathway, so are enhancing, maybe working together the same pathway in the same way. And just to remind you, what is the alkaloid biosynthetic pathway? This is the pathway in the Catharanthus rossus, so in the periwinkle plants. In yellow, you see the final gene that is a DAT. And that is the gene that is regulating in upstream the production of inulin. Catharanthus rossus plants, so periwinkle plants, are important for medical purposes because they produce more than 150 alkaloids. And some of them, like vindolin, vinblastine, and vincristine, are used as an anti-tumoral. So the importance of this work is can be led to the production of these alkaloids for medical purposes. So we know already that again the periwinkle are of great value from medical point of view. So PAV was confirmed as a resistance inducer, enhanced some key enzyme in the phytolexin pathway in both healthy and phytoplasma infected shoots, and the finding that in the infected shoots with PAV, we got an overexpression of CR gene can be interesting for future research because this plays an important role in the regulation of indolin. And this is a promising, I think, use of this solution, not only for preventing or like a resistance inducer, but also for the production maybe of these important alkaloids in plants. And again, thanks to all my colleagues that collaborate in this work, and thank you for your attention. Thank you.
Thank you, Nicoletta. So maybe one question. Question, please. No? Nobody? I saw something raising his hand. Are there online guests? Obviously not.
No. Okay.
So as we are already a bit late, we...
Okay.
Thank
you, Nicoletta. Okay. I invite directly Sofiat Salam. Sofiat. Okay. Because Morteza is absent, so we can move directly in optimal management of witch's broom disease of Lyme in presence of extreme environmental factors using stochastic dynamic modeling. Thank you, Ali.
Good afternoon, everyone. This is Salam Sofiat, and I'm currently completing my master's program in natural resource economics here in SKU. Today, I will be presenting part of my work, which is titled Optimal Management of Witch's Broom Disease of Lyme in the Presence of Environmental Factors using Dynamic Bioeconomic Modeling. Okay. Throughout this presentation, I will be covering the introduction, literature review, model formulation, data sources, model results, and lastly, conclusion. Among the Arab countries, Oman is the largest grower of lime. This is due to the export potential of the crop. In 1970s, the lime industry witnessed experienced the incidence of a devastating disease called witch's broom disease. And this disease presence and spread is aggravated by climate conditions. And for the past, for these years, it has claimed more than 500,000 of the lime trees grown in Oman. As a result of this, Oman government put in place several programs to mitigate the effects of this disease on lime production. These include citrus disease indexing, production of seedling from disease-free mothers, and subsidize the treatment cost for lime growers. All these programs will never result into the desired, will never give the desired outcome until a well-informed decision is integrated into the project. Hence, the objective of this study. is to develop a dynamic bioeconomic model to derive the optimal management strategy for WBDL and the optimal replanting age of citrus trees. This will include the determination of the optimal treatment frequency, timing as well. The second objective is to investigate different cases of WBDL presence and its spread in the presence of climatic conditions. The contribution of the study is important because it is the first to attempt to model and address economic loss resulting from WBDL attack, as well as economic gain resulting from implementing control strategy. The second one is to provide a detailed and thorough economic analysis of the influence of climate factors on the disease and the tree alike. And lastly, this study is among the few that uses a dynamic bioeconomic model and analyze agricultural resources under the threat of diseases in Arabian countries. This literature review, I will go through it briefly, and it is basically divided into two. We have the studies conducted from crop science and that of economics. Considering the ones done from the crop science point of view, we have witch's broom disease and other diseases such as HLB and the like. We have disease and citrus disease management done by those researchers. And lastly, we have the effect of climate conditions on WBDL. All these consider the disease from crop point of view. But turning to economic part, we have few studies on bioeconomic models of disease management. This is done internationally. However, if you consider here in Oman, non-bioeconomic models have been developed in order to mitigate the effect of WBDL or WBDL. I will go through this model formulation quickly. Basically, we have 4 continuous variables and a binary state variable. The first of the continuous variable is climate change, whereby we look at the index of climate change. We assign weights to each climatic factors, and we consider the base value for each region. Then the second continuous variable is the vector density and disease propagation, whereby we see that the density of pests on a field will be determined by the density from previous month and the growth, which is impacted by climate condition. Then the disease density alike. The same thing goes for this, just that we have the action of the farmer to control the disease or not. He can control it or decides not to control. Then we have citrus growth function, which consider the biological tree growth. And also, we have the growth, which is impacted by disease and density and the climate condition. Then after the impact of the disease on lime trees, we have salvageable value. That is one captured by the last part of the equation. This one gives us the net benefit that will be gotten if the farmer effectively implements the control strategies. That is, we are trying to maximize the current and future benefits of lime using this equation, this consists of different, we have the price and the growth of the tree. Then we deduct it from those costs. Then we had the discounted future net benefit. So the study uses climatic data and as well as citrus and disease data. Climatic data was gotten both internationally and locally. Then citrus and disease data were collected from citrus growth data survey, field data survey, and direct observation. We got these parameters and specification looking at the literature and also by author's calculation. These are the second column shows the functional form that will be used to calculate the fruit mass growth, vector growth function, and the rest. So turning to the result part of this study, we look at the relationship between the age of tree and the fruit mass. We find that there is a significant relationship between them, and the model is statistically significant overall. Then this study uses 6 different scenarios. The first scenario is the case of a healthy tree. The second one is a tree that is infected by WBD and then no treatment. That means the farmer does not engage in any treatment and strategy. Then the third one is the case of infected tree but treated. This first 3 scenario is similar to the last one, just only that we added climate condition to the last 3. And this is the preliminary result of the study. Here, the first column, we see the expected site value, which is the expected economic value that the farmer will gain in these different cases. The first one is the baseline, where we have the case of healthy tree. This one gives us as much as 69,000 Omani Riyal. If the tree is healthy, no disease, everything is working fine. However, once the tree is attacked by WBDL, the expected economic value drastically reduced to almost 9,000 Omani Riyal if the farmer does not undertake any control strategy. In case the farmer chooses to control the disease, then he is going to get around half of what the LV tree will give him. This is around 35,000 Omani Riyal. So looking at the optimal replanting age of the tree, for the healthy tree, it is optimal to let it live for up to 35 years. Whereas in the case of an infected tree that is not treated at all, the tree it is better to cut the tree once it reaches the age of 9. It is not even up to half of what is obtainable in case of healthy tree. Now, the last most important thing here is that disease treatment frequency and intensity matters a lot if and only if we want to achieve the desired result of controlling this devastating disease. So the disease treatment frequency is 20 altogether, and it should be carried out weekly during the harvest season, that is May, June, July, August, and September. So if this treatment strategy is employed effectively, then the farmer that is witnessing the problem of WBDL is going to get half of the LD tree. Then we move on to the disease treatment intensity, whereby the farmer can decide to, the farmer, if you want to optimally manage this disease, must prune or rogue all the diseased branches in the middle of the harvesting period. However, you can decide to reduce the amount of roguing or pruning to 93 or 91% in May and September because it is costly. So this case that I just described is without considering climate condition. And we all know that in agricultural production, climate conditions do matter a lot. So in the second result, we introduce climate condition. Here, we have the expected site value to be reduced compared to what is obtainable when we don't consider climate at all. We can see that the rotation age also, that is the replanting age, also reduced when we integrate the effect of climate change in the model. And also, honestly, this is what is happening in real world, because agricultural production cannot happen without climate. So this result is corroborating other studies done by previous researchers, which state that environmental factors such as temperature, humidity, especially humidity, are very impactful in the productivity of agricultural crops. So now the disease treatment frequency is still the same. We have to prune weekly throughout the harvesting season. And if we turn to the disease treatment intensity, we see that there is a difference between what we have in the case whereby we don't consider the climate change. We have to prune all the disease, the symptomatic branches in order to achieve the desired result. Conclusively, the disease attack and climate conditions are important factors that increase the resulting damage of citrus fruits in Oman. The farmers' pest management decisions are significantly impacted by the pest and the disease density level, and climate conditions play a major role in citrus management. Farmers should account for the biological growth features of the pests and the disease on the farm throughout the season before making any management decisions. Farmers should also undertake an increasing level of frequency of the disease treatment throughout the whole season. It should be noted that the current disease management actions by farmers are not sufficient, because here in Oman, they usually implement the control strategy only in September. That is at the end of the harvest season. Based on the results of this study, it has been shown that this control strategy is not enough. In order for them to optimize their return, they have to increase the frequency and the intensity. And thank you for listening.
Dear Sofiat, many thanks for very interesting presentation. Are there any questions? Question for the web? No question. Okay, many thanks, Sofiat.
Thank you.
So we come to the next talk, enhancing the survival of sugarcane infected by white leaf diseases using bacterial indolactic acid producers, and will be presented by, I hope I pronounce this correctly now, Kok Bao Nguyen, approximately.
Yes. Okay, so good afternoon, everyone. So first, also, I'm very happy to be here for my talk involving the small project in my research, and this is the first time to be in Oman. It's a beautiful country. So my talk focus a little bit about the application of, not application, that the initial evaluation of PCP, like plant growth promoting bacteria. So we use like IAA producing bacteria, and we check if it works to recover that the phytoplasma infected plant. So in Vietnam, so we focus about that the sugarcane wild leaf disease. The sugarcane wild leaf disease, so is the first observed in Thailand, in the Lumbang province, in the northern part of Thailand, and it has become outbreak disease and caused severe damage of the sugar industry, not only in Thailand, but also in Vietnam. And in Vietnam, so the first discoveries of the sugarcane wild leaf disease, so in the south of Vietnam, and then so it spread, the disease was spread to many provinces from the north, from the central north, and the highland region, and the south of Vietnam. Mainly it's got a severe loss in the sugarcane growing area, the biggest sugarcane area in Vietnam. So about the description of a sugarcane wild leaf disease, of course, so we can see that the symptom of the sugarcane wild leaf disease or sugarcane grassy shoot disease or green grassy shoot disease or ramustone disease is caused by the phytoplasma. And in Vietnam, so normally, so we only see that the sugarcane wild leaf disease and sugarcane grassy shoot disease. Sugarcane grassy shoot disease, so we observe this is mainly in the north, in the central north Vietnam, but in the south, like Tây Ninh province or in the highland region, mainly, so we see that the sugarcane wild leaf disease. Previously, so we also contributed in one chapters, book chapters, and we focus about that. We indicated some management approach in Vietnam to control sugarcane wild leaf disease. For example, so we try to set up that the system of production and propagation of healthy sugarcane. So we have that the 3-tier 6 program. Under that, the guys from the Sugarcane Research Institute and Vietnam and the Ministry of Agriculture and Rural Development in Vietnam. Besides that, so we also established many training courses for the farmer to see that's the dangers of the using cutting from sugarcane wild leaf disease for breeding. And we try to identify the vectors transmitting phytoplasma, and regularly visit the sugarcane field to discover and destroy sugarcane wild leaf disease infected root cane. And particularly, so we also evaluate that the tolerance or resistance of the sugarcane wild leaf disease for on domestic and imported sugarcane variety. But actually, it's really hard to control the disease in Vietnam. So sometimes some suggestion that we try to minimize import for foreign varieties. Normally, in Vietnam, so we prefer that some sugarcane variety from Thailand and Taiwan. But unfortunately, so most of them, they are the, phytoplasma susceptible variety, even though like it's provided the high yields and high quality. And so now only one way to control the sugarcane quality disease in the fields is so we remove or cutting from the infected area, or we burning in the fields. In another approach, so we are thinking about the roles of the plant growth promoting bacteria, because as you know that so many papers they only indicated that the functions of the BCPP like to improve the plant nutrition or regulate the phytohormone, or to reduce or prevent infected plant. So I just think that the symptom caused by phytoplasma because that the effect of that phytohormone disorders from the phytohormones change in the plant and cause that the symptom, the phytoplasma associated symptom. So in this case, so we try to isolate and identify some bacteria, II producing bacteria. So we collect that the rice root, because in rice root so there's many endophytic bacteria. So we isolate, and then so we observe like under microscopy, and we evaluate that IA production from the bacteria isolation. We identify at least 7 bacteria isolates can produce IAA, and including the 4 gram positive and 3 gram negative bacteria we identify. And so we identify at least 2 bacteria isolates produce the high level. So we use 2 bacteria isolates for further study. And of course, so we try to identify that the specific name using cysteine ribosome RNA. And then so we identify 2 bacteria isolates so that we can use for our experiment is Diphtheria lacustris and Ranella aquatilis. So we just test if IAA producing bacteria can stimulate, enhance the growth of the plant. So we use the rice cultivar. So we use like the hybrid rice and the local rice variety, and the result indicated that so it's that the rice treated with the IAA producing bacteria showed higher than the control, even though the plant grow and also that the root, the length of the root. Based on that result, so we thinking how we can evaluate in the sugarcane sugarcane quality disease infected sugarcane. Before to do that, so we use one variety, a sugarcane variety from Thailand, a Khon variety. We call it KK3 variety. So that is a phytoplasma susceptible. It's very easy to be infected with phytoplasma. And we try to evaluate that if most of the materials has the infection with the phytoplasma. So I have a collaboration with Professor Matthew Dickinson from Nottingham University to develop that the LAM-based assay. So very quickly, so we use that the LAM assay that to detect for screening, and we just use that the infected plant for the study. And we already tried to develop that the LAM primer to detect only sugarcane wild leaf disease because before we use like normally so Matthew use the primer, LAM primer that can detect Napier grassy stun So that primer can detect Napier resistance and sugarcane leaf disease. So we try to develop that specific primer that only detect sugarcane leaf disease. So very easy to screening, and we select that infected, and that's a broad can use for the early detections of phytoplasma. And we use that strategy developed by the Professor Matthew Dickinson, and we do the screening and to detect that the sugarcane leaf disease in Vietnam. So actually, so when we use two IAA producing bacteria, so we see that the significant change about that the height of the sugarcane between the control and the treatment significant. And the interesting thing is, so we see that the symptom of the wild leaf disease, the symptom of wild leaf disease, there's a difference. We see that the efficient reduction of the symptom of the wild leaf disease with the treatment of the IA bacteria, producing bacteria. And we also see that the survival, you know that, so the sugarcane infected with the phytoplasma is becomes stunted and die at the sickling. But when we treat with that IA producing bacteria, so it will help that the plant recover and grows very high, and the most important is that we didn't see that so much symptom, still have, but not so much symptom like the control. That's quite interesting, and we don't understand that the mechanism. I think that's very interesting to explore the mechanism, but we try to use those bacteria to control the cassava mosaic virus, and it's sort of similar. I don't show the result here, so we can see that the significant reduction of the mosaic the symptom caused by the mosaic virus in the cassava. Anyway, so I think it's very interesting, so to understand to find out that mechanism. So what happened when we do that? Even though like IA producing bacteria or some bacteria involved in that the nitrogen fixation of phosphorus soluble solution, so also called that the significant reduction of the disease. So maybe there's some mechanism. Maybe in the bacteria they also have some compound that's inhibit that the replication of the phytoplasma or virus. I just jump to the conclusion that at least so we identify two bacteria, IA producing bacteria that providing oxygen and other substance to assist phytoplasma infected sugarcane seedling to overcome that physiology disorder. And we can see that the enhancement of survival ability of sugarcane seedling at the early stage of infection. And of course, the PRPs were not able to completely resist phytoplasma. Phytoplasma is still there, but the thing is, so like in the cassava mosaic virus, so we see when we do real-time PCR, we see the significant reduction of the virus in the plant treated with the PIP. I'm sorry that I have not checked with the phytoplasma by real time, but I think that I think that there's the reduction of the phytoplasma concentration in the treatment. So, but it had ability to delay the onset of severe disease in sugarcane seedling. So that is the conclusion in our research. Yeah, so finally, this is a shock. I would like to take a little bit time because my work focused mainly about that molecular diagnostics in DC. So in this December, so we organized that the international conference about the molecular diagnostic in microbiology in DC, held in the center of Vietnam. We know this beautiful landscape with the sea and mountain. So if you're interested, you can join that event. This is the second time. It's a rencontre du Vietnam. We organize in ICISE and Quy Nhơn. And finally, so I would like to thank Professor Matthew Dickinson. So he's the first person joining the first project funded by the World Bank and the Ministry of Science and Technology. So he helped us about to develop the lamp safe for detect phytoplasma cause disease in sugarcane disease. And thank you so much for your attention. I'm very happy to receive any question.
Thank you for this interesting approach. Are there any questions?
Thank you for your interesting presentation. Yes, there is some space for this way to reduce the concentration of the phytoplasma, but this can be more dangerous because you see asymptomatic plants that are still infected, and you have the insect vector. So this is not a solution. It's just a mitigation, should be not taken as, something to apply directly like this. You can do if you do not have a vector, but I am aware, at least in Thailand, I don't know for Vietnam, but I think you also have an insect vector.
Yes, there's the insect vector, but unfortunately, so nobody identified that the vectors called the sugarcane disease. But I think that's similar, like in Thailand. I think similar to Thailand. For the application of the IA producing bacteria, so that's the big question. So we thinking how we can do, because how we can control so many factors in the field that we can, reduce the loss caused by the phytoplasma. Yes.
There were two other. John or.
So I have one comment and one question. So I'll go with the comment first. Sugarcane phytoplasma is known by both of them, sugarcane grassy shoot phytoplasma and sugarcane white leaf phytoplasma. You have used third term SCWLD. Okay, just a suggestion. I mean, it has been proved by genetically that the phytoplasma strain which infects sugarcane is the same. It's identical in genomic nature. So somehow it is like we use SCGS oblique SCWL. So this is to reduce the confusion, because if somebody sees your presentation now, you're talking about third phytoplasma other than SCGS, SCWL. You also mentioned in your presentation that SCGS and SCWL are different phytoplasmas. So maybe there has to be some relooking is required, I believe. Maybe we can discuss it later. But there are multiple papers which says that both the phytoplasmas are same. My question now is, what is the source of these bacteria that you have used? And if I'm not wrong, these belong to gamma proteobacteria, and both of them, Delphia and Rhamnella, are may not be these strains, but they are pathogenic in nature. To human pathogenic they are. So they may not be active, that particular strain, but they have same genomic nature, and we know that the opportunistic bacteria, they may remain virulent until unless they are not have an opportunity. But using these, have you checked their any virulent protein or any virulence of them?
I'm sorry if I interrupt you, because we must go on.
Yeah, right. So my question is, what is the source of those isolates?
Yes, unfortunately, so I think that's one of the bacteria that could be not good for the human. But the thing is, like we try to use another like bacterial isolates like nitrogens and phosphorus soluble bacteria. Though we also have a two strain. It's safe for the human. It's the benefit bacteria, and it also produces the IAA, and I already test, and it works well for the reduction of the disease, not only phytoplasma, but also in the virus. And you know that some bacteria, even though they produce the highest IAA, but actually so we cannot use in practice. Yeah.
So a last short question, please.
So my question is, did you add
IAA in your treatment without bacteria?
Not yet, yes.
Because I couldn't see if you correlate between the bacteria produce IAA, so I would recommend to use these first to see that because of this IAA is help this treatment.
Yeah, thank you. Thank you for your recommend. Yes.
Thank you very much. So we come to the next presentation.
Okay. We can move with the talk production of sugarcane grass shoot, disease-free, set by using hot water treatment. The talk will be presented by Ayak Kumar Tiwari.
Thank you IPWG for this opportunity to be here with a number of leading researchers of the phytoplasma related to phytoplasma disease. This is A.K. Tiwari from India. My topic is related to disease-free nursery of sugarcane, which we produce and distribute to the farmers. As the topic is related to sugarcane, which is known as for the sweetness in our life, our day start with this sweetness. So the sugarcane, the Brazil, which ranks first for the sugarcane production, and India, from where we belongs, is ranks second among the worlds. If we talk about the Indian scenario about the sugarcane production, so around 5 million hectare area under cultivation of sugarcane, and we produce around 400 million metric ton sugarcane, which if we talk about the Indian continent, the two main sugarcane growing areas in the world, India, that is tropical and subtropical, we can divide it. And the southern part belongs to the tropical belt, which is contributing around 2 million hectare area. And the most important one in the subtropical belt, from where my institution belongs to, which is contributing around 3 million hectare area under sugarcane cultivation. We have 525 sugarcane factories in operational. When we talk about the subtropical part of the India, so the states Uttar Pradesh, which is contributing around 3 million hectare area, 2.7 million hectare area under sugarcane cultivation, and where 119 sugar factories are operational. If you see the last 5 year cultivated area of sugarcane, then you will find that which is around 2.7 million hectare area, and it is in an increasing order, gradually increasing. Our institute is located in the center part of the state, and our state Uttar Pradesh is also divided in 3 parts on the basis of climatic zone, that is the eastern, central and western part. Our institution is located in the central parts, and we have a total 10 centers across the states. 6 in eastern part, 2 in the central parts, and remaining in the western parts, only for sugarcane research and distribution, multiplication of the breeder seed and distribution to the farmers. So we know that sugarcane is an annual crop, and throughout the year it faces number of diseases. From India, around 60 diseases has been reported on sugarcane. Out of these, major diseases are economically important, and the most important disease of the sugarcane is caused by the fungus, that is the red rot disease. And most of the diseases of sugarcane are seed transmitted diseases, which are the major ones that are spread by the seed, that are seed transmitted diseases. And if we go through the literature, then we will find that losses caused by the disease range from 50 to 20% annually. But in some cases, we have 100% yield losses, like as I mentioned, the red rot disease. Recently, in last 3 to 4 years, we have seen the epidemics of red rot, and we lost the wonder cane of that is a CO0238 variety. Number of fields we have surveyed, and there was we did not find a single healthy cane in 1 hectare area. So there are some diseases which are causing 100% yield losses to the sugar canes. But what after the fungal diseases? As I mentioned that fungal disease stands on the first rank in a subtropical belt of India. We don't have much incidence of bacterial and viral diseases, and not significant losses are there due to these diseases. But we can say that phytoplasma disease of sugarcane stands second after the fungal diseases. And number of work has been done from India and also some Asian country, and also from some Asian countries. And during our literature, we find that the workers reported less germination due to the grassy shoot disease, high reduction in yield, poor ratooning. From 2008 to 2012, we done a deep survey because of epidemics of grassy shoot disease, and we find number of fields were found affected with grassy shoot disease, and there was up to 40% qualitative and quantitative losses in case of standing cane. But if we have a green grassy shoot disease which having no cane formation, there is a chances of 100% yield losses. Here I have shown you some pictures of the grassy shoot disease. The first one is the healthy, and you can see the symptoms of grassy shoot disease. The most interesting one are the below left side. You can see green grassy shoot disease. No cane formation is there, grassy appearance, and 100% losses is there. And the last one is the yellow leaf disease. Some reports are there that yellow leaf disease are caused by the viruses. However, we have confirmed that it is also caused by the phytoplasma. So the disease caused severe losses to the malleable canes, and the severity of increased multifold in the ratoon crops, resulting into a complete reduction in cane yield. The use of healthy and vigorous seed plays a vital role in the cane development program, and seed is the main constituent of all the inputs required for good quality and yield of sugarcane crop. We know that sugarcane requires lots of seed materials, like 6 to 8 tons seed materials for 1 hectare area, and the seed sets are the costlier inputs, which accounts for nearly 25% of total input of production costs. So planting of infected seed materials which may carry latent infection within the tissue are largely responsible for an annual recurrence of diseases. Sugarcane grassy shoot, or you may say wild leaf disease infection, results serious reduction not only in cane yield but also in the juice quality. So we have a only solution till now is to produce healthy seed cane material. We know that we don't have any chemical remedy to overcome from this situation. So we can go for either producing healthy seed nursery through tissue culture techniques, but it is costlier, or by going to heat therapy. The heat therapy is not new to us. It is earlier done by Qaboos in 1989, and from India, from our institution, our ex-director in 1978, he used the heat therapy to remove the albino. The grassy shoot was earlier known as albino disease also. In 2001, Dr. Viswanathan from southern part of India used aerator stem heat therapy to remove the grassy shoot disease. So this is, you may say, our regular feature program to produce grassy shoot disease-free sugarcane seed materials and this distribution for foundation nursery. What we have done, that we used the most popular variety at a time, 08272, with the symptom of grassy shoot disease. We used two temperature, that is 50 degrees Celsius for 2 hours and 52 degrees Celsius for 1 hour. And the second segment of treatment, we utilize only 50 degrees Celsius for 2 hours for planted along with control, that is symptomatic, and also planted without hot water treatments. And only we have taken germinations and symptoms of the data. These are the hot water treatment plants. We have used it for 50 degrees Celsius for 2 hours. Sugarcane seeds were placed inside the cane basket and lowered into the water tank. Each basket, there are 9 baskets is there. Each basket can contain 40 to 50 kg sugarcane materials, and maximum up to 5 quintal seed materials can be treated at a time. These are the results. We found that 50 degrees Celsius for 2 hours is quite better because it having a good germination, and 50 degrees Celsius per 2 hours is also better, but it having less germination as compared to 50 degrees Celsius for 2 hours. So we proceeded with the 50 degrees Celsius for 2 hours. And 3 consecutive years this experiment was done. Not you may say experiment, this, we have run this program and found that 50 degrees Celsius per 2 hours is quite better because it having a good germination, less incidence of the disease. So we are regularly, we are doing at all our center to producing disease-free nurseries. As we have a number of farmers, we have around 4 million farmers in our states. So we have to meet out the demand of sugarcane seed materials. We produce only breeder seeds at our centers, and these breeder seeds we distributed to the sugarcane factories, farms for the foundation of for foundation nursery. And during breeder seed production program, we do at least 3 visits to certify the seeds. The most important visits are before the harvesting of the cane. At this time, if we found a single incidence of any disease, like red rot, grassy shoot disease, smut. In case of red rot disease, we reject complete field. If there is a single cane which is infected with the red rot, we suggest to send all the materials to the sugar factory. But in case of grassy shoot disease, smut, and wilt, we suggest to rogue out this, and strictly recommendation are there to use hot water treatment before using it as a foundation nursery. So annually 1.5 lakh quintal seed ton materials we produce and send to for the foundation nursery. Here what I was saying about the seed certification standards. You can see that the tolerance level of the disease at breeder seed level, red rot, zeros, smut, GSD, wilt, we do not tolerate this one. Strictly we follow that standards. So when we have epidemics of red rot at in our states, so how we can multiply the newly released variety at a faster rate? We know that we can multiply through tissue culture at a very faster rate, but it is very costlier. So we suggest farmers to use a single space transplanting method. It having a less requirement of seeds and faster multiplication because it's having a good germination percentage. And when you have a good germination, you will have also a good productions. Sometimes it happens, people ask us to how you are communicating to the farmers. You have a number of farmers in your states. So we have interestingly, I must say that we have a Facebook live program in YouTube programs, which having a 1 to 4 million reach. You would not believe, we having a 1 to 4 million reach through Facebook live program. So whatever we have to communicate to the farmers, we just, on every Saturday at 4 p.m., we just do the Facebook live program and communicate to the farmers. So we suggest to the farmers that if you can't able to get breeder seed materials from us, by the way, any reasons, if you are not getting, so you can prepare your own seed materials. How you can he can prepare at least 10% of seed materials what you are using must treat with hot water, and these use every year. So you will have a healthy seed materials, because as I mentioned earlier, unless we don't have any chemical remedy for grassy shoot disease, we must have use healthy seed materials. So that's all that what I was mentioning in my all the slides, that hot water treatment sets before treating 50 degrees Celsius for 2 hours to mitigate the infection of grassy shoot or white leaf disease is suggested. And the 3-tier seed multiplication program has drastically minimized the incidence of disease. And because of this, we have a 82.5 ton per hectare yield at our average yield of our state, which is highest one in the last 10 years, you may say. So in last, I must say thank you to TSBS for the financial support, Professor Abdullah, Professor Subi for their invitation and, and support. And I must say, Chair, the Professor Sunta, for their invitation and everything. Most of the times he supported us. Thank you.
Thank you. There is a space for a very short question. Otherwise, I kindly ask to do during the coffee time. Okay, many thanks.
Okay, so we come to our last talk, control methods of alfalfa, which is broom phytoplasma disease, which will be presented by Syed Alireza Esmailzadeh Hosseini.
Good afternoon, everybody. Our presentation is about control methods of alfalfa, which is borne phytoplasma disease in Iran. Alfalfa is called green gold and queen of forage plants. Is very important in Iran, Middle East, Central Asia, and particularly Iran as Turkmenistan. Due to importance of alfalfa cultivation in Iran, cultivation of native cultivar in different areas of the country, and compatibility of different ecotypes of alfalfa with different climate region, study of native alfalfa and the importance of seed productivity is necessary. In recent years, we have different phytoplasma disease reported from Iran, like pistachio, which is boron, sugar beet, and garden beet, which is boron, sesame phyllodes, cucumber phyllodes in the farm and in the greenhouse cucumber, carrot and other phytoplasma disease, and also alfalfa, which is boron as an important alfalfa disease and phytoplasma disease in Iran. The total number of damage to alfalfa crops is estimated to be 300%. In recent years, alfalfa, which is boron, has been one of the most important factors in reducing the quantity and quality of forage and seed production of alfalfa. These symptoms are similar symptoms of phytoplasma disease are clarified in the next slide. For the first time in 1925, alfalfa, which is boron, were reported in USA. And in Iran in 1993 was reported in Fars province in south west of Iran, and demonstrated based on biological and molecular studies. The disease is so important that in some part of the country farmers were plowed due to alfalfa disease damage and use another product like pistachio. In 1999, epidemics of alfalfa, which is boron, occurred in Chahgir in Yazd province, and farmers reduced and the damage of the alfalfa reduced to less than 1/3, and seed production was not possible for the farmers. Now alfalfa, which is boron, is distributed in all parts of the Iran with different disease incidence and severity. Based on symptoms and symptoms showed by the infection, we have 5 disease severity, healthy, and 4 disease rate, as you see in the first stage, and also died of the plant, infected plant. Healthy farm, in the flowering stage you see a healthy farm, and also whole infection of farms completely infected by the alfalfa, which is boron phytoplasma disease. And death of infected plant in the summer and also in the winter. And we saw heterogeneity in death of infected plant. As you see, different plants with this different disease severity died due to infection with alfalfa, which is boron disease. And a 3 years stand alfalfa abounded due to crop loss of alfalfa, which is boron. Healthy plant and infected plant, in the healthy in the left and the infected in the right. As you see, seeds from healthy plant and also in the infected plant. There is no report in Iran about seed transmission of the phytoplasma. And two important vectors, the left circulifer hematocells and the right Orseus albicinctus. And the necessity of conducting this research is direct damage to alfalfa in the forage alfalfa and also in seed production, and also important role in the epidemiology of the phytoplasma disease and infection of to other plant disease. The strategy applied in this research, we use different methods of alfalfa disease control in a randomized complete block design and tree replication. The methods were dense planting on alfalfa, use of sorghum as windbreak, and spraying with metacystox R. And why these control methods were selected? The first method, dense planting of alfalfa, farm infection usually start at beginning and border and tiny area. It is reported by Menzies in 1951, and density of planting was evaluated for the potential to control of alfalfa, which is boron. As you see, in farm boundary, the rate of disease incidence is very high in this area, and also in the place of water entering in the farm, you see a traditional water irrigation. The incidence and disease severity is very high in this area.
Sorry, but you must speed up a little bit. We are already 15 minutes late, please.
Yes, yes. Dense planting, 25 kilogram per hectare, and also twice it, and use of sorghum as windbreak, 3 layers and 3 rows of sorghum around the plots. As you see, sorghum by color, and spraying with metacystox R for the killing the vectors. Disease incidents were used by sampling in a plots and molecular studies. The infected plants show elongated flower malformation and sterility. Morphological change in the infected plant, healthy plant and also infected plant, virescence, and infertility of flowers, dwarfing and increasing in the leaf numbers, dwarfing and smooth leaf age of infected plant. Infected strain of phytoplasma is a peanut witches boron subgroup D. And you see in the phylogenetic tree, after application of different methods, only dense planting reduce alfalfa witches boron control by 16% compared to control, and metacystocks and use of sorghum as windbreak do not affect in reducing the disease rate. And you see in a very thin field or a sparse field, rate of disease is very high, and dense planting is very effective in control and preventing infection of the disease. With increasing the amount of density, disease incidence and severity increase. Until now, no cure methods were identified for alfalfa witches boron, but a different rate of symptoms and field resistance or tolerance were observed in the field. Some plants in the field show disease symptoms with high disease severity, and also adjacent plant next to the infected plant has healthy or without any symptoms, like this. And seedling from healthy in the left, and the right seedling from the infected plant, low vigority and high vigority, and comparison of alfalfa seeds from healthy and infected plant, despite any report about seed transmission. We don't have any report of seed transmission of alfalfa witches boron in Iran, but the vigority of seeds from infected plant is low. As alfalfa is perennial, no cure methods is available. Use of density is not completely effective, and due to lack of disease resistance, alfalfa cultivar ineffective methods of windbreak and spraying with insecticides and other cases. Alfalfa witches boron phytoplasma disease control is difficult and requires the use of combination of different control methods. In the area where alfalfa disease are widespread, was likely to be present in addition to management aspects such as destroying highly infected farm and transporting alfalfa as dry. Farms should be planted with high density and good agricultural practice. Considering that condition of the soil and number of type of irrigation water have an effect on the amount of seed used, the best alfalfa planting density for reducing disease incidence is different each region and should be tested locally. Thank you very much for your attention.
Thank you very much, and for saving time at the end. Thank you. Okay. So, one question. Everybody's tired now? So, thank you once again, and I will close this afternoon session now. I thank all the speakers and also the auditorium for the patience, and I think there will be an announcement now concerning this dinner for tonight by Ali. Thank you.
Thank you very much, Barbara. Thank you very much.